phytoplankton abundance, zooplankton abundance, diversity, and light response in the Gulf of Santa Elena. And I am sorry if I say zooplankton, I just think it's more fun. All right, to get everyone on the same page, I'm going to give a little background. So what is plankton? Plankton are aquatic organisms that drift with water currents because they are unable to swim against water currents. And they are the base of our marine food webs. They are the lowest trophic level, and they are eaten by larger marine organisms. And they are incredibly important for our biogeochemical cycles because they are a large part of our carbon sink and also part of our nutrient recycling system in the oceans. And you might have heard of phytoplankton and zooplankton. So the difference between these two is that phytoplankton are autotrophic, which means that they are primary producers and they conduct photosynthesis to turn light energy into chemical energy. And then zooplankton, which is the plankton that I looked at, are heterotrophic and consume other microorganisms like phytoplankton for energy. Okay, great. So to get us all on the same page, I'm going to be talking a little bit about morphospecies throughout this presentation. And morphospecies are very similar to species in their grouping, but because I can't identify these organisms down to their species level, I will group them in morphospecies, and they have different taxonomic classifications. So for example, Decapoda is a order, while Mollusca is a phylum. And this just makes it easier for me to group my identifications together. And finally, I mentioned light responses in the title. Light is very important to our marine organisms and is a limiting factor for primary producers, and therefore a limiting factor for zooplankton. And zooplankton are also very well studied for their vertical migrations throughout the day. What they're known for is swimming up the water column at night to feed on phytoplankton as an adaptive measure to avoid predators during the day. And then additionally, light wavelength and UV radiation can influence coloration and distribution of marine organisms, and different wavelengths of light are scattered and distributed differently in seawater. And so to get to my central research questions, my first one is, how does zooplankton morphospecies abundance and diversity differ across marine ecosystem sites in the Gulf of Santa Elena? And then my second question is, how do morphospecies differ in their response to wavelength-specific visible light and UVA light exposure? And the study sites that I was looking at were coral reef, boat docks, rocky reef, and open ocean. So to conduct my study, I would go out after sunset with a plankton net, many plastic bags filled with alcohol, flashlights, and a dream. And also a very nice team who helps me as well. And so at each site, I would conduct plankton tows, vertical plankton tows down to 5 meters. And I would do this for one dark condition, so just without any light. And then the next time I would put in a UV flashlight in the water, like just below the surface for 10 minutes, and then take samples, and then I would repeat that with green light and then white light as well. And then after collecting my samples, I would look at each sample under a compound microscope 5 times, and these are called slide observations, and that's how I decided to catalog all the zooplankton morphospecies. All right, and now to the best part, results and discussion. In total, I identified and counted 2,153 individual zooplankton, and they are categorized into 20 distinct morphospecies. And these morphospecies range from everything from like phylum to suborder, superorder, class, and even family. And this first figure is complicated, but I will explain. No worries. It's about the occurrence of morphospecies by location. So in the x-axis, you will see that they're separated by the 4 marine ecosystems that I was talking about. And then on the left-hand side is the morphospecies code. And basically, like CPD would stand for like copepod, or OST stands for ostracod. And then on the right-hand side and in the cells, you'll see just the like actual numerical abundance of the organisms. And so from this figure, we can see that the coral reef has the highest number of total zooplankton, and then also has the highest number of copepods. And then next up is boat docks, which has the next highest abundance of zooplankton in total, and also copepods. And then rocky reef has the third highest abundance of total zooplankton abundance, but the least amount of copepods. And then open ocean has the least total zooplankton. And what we can gain from this figure is that coral reefs have a very high number of zooplankton and phytoplankton, and this can possibly be attributed to the fact that coral reefs are often found in very sunlit places, and that they're known for having a very high productivity. And because phytoplankton needs sunlight, this results in more photosynthesis and more zooplankton as well. And then also another point that I want to add is that the coral reef study site that I was looking at had the shallowest depth out of all the sites. So that might have been a factor because I mentioned like some zooplankton move up through the water column. They might have been more concentrated, and therefore I would have gotten more of that sample. And then boat docks are close to human activity, artificial light at night, and are subject to a lot of pollution or like extra bioorganic material from fish that's like thrown off the docks. So that could be another reason why boat docks have the second highest abundance of zooplankton. And then open ocean might have the least because it's furthest away from any human activity and furthest away from the coasts, and just also the deepest depth that was of any study site. Okay, moving on. Copepods were the most abundant morphospecies in the boat docks, coral reefs, and open ocean sites. And this actually tracks with historical research because copepods are some of the most abundant animals in the world, if not the most abundant. And they dominate zooplankton in marine and freshwater environments, and often are like 50 to 90% of zooplankton in the ocean. Okay, next, let me speed up because Frank's looking at me. The total zooplankton abundance by condition is a very nice trend. It's clear that when any light treatment is applied, there is an overall increase in zooplankton, and this is a total of all of the morphospecies that I counted. And the white light has the most dramatic increase at about 54% compared to the dark condition. And then next, this is a similar figure to the first one, where I'm looking at occurrence of morphospecies by condition instead. And we can see here that the white light condition has the highest number of morphospecies, also known as like highest species richness, and then also has the highest number of copepods here. And then the other three are quite similar in their species richness, which is interesting, unexpected for me, but it does make sense because zooplankton are unable to swim against currents. So if all of these study sites are in close proximity to each other, it makes sense that they would all have similar morphospecies. And then moving on, white light had the highest abundance of copepods, and UV light had the highest abundance of cyclopods, which is an order of copepod, and then green light had the highest abundance of ostracods and radiolarians. And this is interesting because while the species richness of 3 out of the 4 study sites were similar, of the conditions were similar, we can look at the specific morphospecies and see how their light responses or photosensitivity varies based on the wavelength of light. And this is something that definitely requires further research. Oh, okay, I'm almost done. Further research because it was really difficult for me to identify species or morphospecies down to the species level, which could be improved with other genetic tools. And finally, average species richness was a way for me to look at which location commonly had the most amount of morphospecies per sample, and what I found was consistent with my findings for species abundance, in that the coral reefs had the highest average species richness, and then also the white light condition had the highest average species richness of that location. Okay, skipping that, thank you so much. Just to wrap up, what we learned was that coral reefs have the highest species abundance, and copepods are the most abundant morphospecies. Coral reefs also have the highest average average species richness, and that different conditions of light can tell us different information about morphospecies photobehavior, which is response to light. And finally, something that I would want to look at in the future and more, it's also in my paper, come ask me if you want to learn more about it, but it's morphospecies specific behavior, looking at individual orders or species or families within a larger morphospecies classification and seeing how their responses differ and how their distribution also differs. Finally, I would not have been able to complete this project without Frank, my primary advisor, and Emmy, my secondary advisor, and thank you so much to everyone who helped with my project on Nightboat or editing or identifying. It was a pleasure and a blessing to spend 2 weeks in Guayaquil doing marine research, and I really loved it. So thank you, everyone.
Do we have time for one question? Yes.
First off, amazing job. Loved it. I learned a lot. And second, what do you think the implications for conservation are, like following?
That's a really good question. I wanted to talk more about that because scientists have been looking at the relationship between plankton and oxygen, and there is a threat of extinction or like decreasing populations for phytoplankton that are providing a lot of our oxygen on the daily. And so it's really important to look at the species distribution and population of zooplankton because they're pretty much like keystone organisms when it comes to bridging that gap between like phytoplankton and larger marine organisms, because if their population like blooms or decreases drastically, that puts so many other species at risk. And there have already been studies in the past, looking at how the geovertical migration of different zooplankton species are changing because of warming ocean temperatures and other factors of climate change. So, very important. We can talk more about it. Thank you, Elise.
So, I'm going to move my mustache so you can see that I'm smiling. Okay, so Luchi, what's the story?
I sent it to you and everyone has it.
Did you send it by email?
Yeah,
Let me see if I can get it.
I said strange.
Luchi, do you have it on your computer?
Yeah.
On your computer?
Yeah. Do you not have an email or you can? I think you want to put
There's no email for me, at least.
You can also airdrop it, Luchi.
You can try to airdrop it if you want.
Go to airdrop and then you airdrop. Okay, Lex. I saw you like.
Vivi, can you help me?
Yeah. I actually downloaded it right there. Okay, so now go to the speaker notes and then go to downloads. And then this is I don't know how it show up. I don't know how this one's custom and then go to get on and then go
So you're up? You're choosing the process.
It should be going to your computer. Yeah.
¿Cómo se dice eso? Okay. who will be talking about nudibranch coloration.
Hello, as he just said, this is my presentation on nudibranch coloration. So a little bit of background info on nudibranchs. First, like where they're distributed and their ranges and where you can find them. They can be on the eastern coast of North America, so you do see these specific species of Elysia diomedia, which is the ones that I looked at like in California. And then they go all the way down to the Caribbean Sea and to the Gulf of Mexico, so all the way down here, which is where I found them also. They're part of the mollusca phylum, along with octopus. I got a little cartoon right there. Squids, clams, snails, and mussels as well. This species specifically that I looked at, the Elysia diomedia, they don't have a hard exterior shell in order to protect themselves from other predators, so they have to get other characteristics and other types of functionalities in order to escape predation. And then lastly, what sets this species aside from other groups in the mollusca phylum is their ability to steal functional chloroplasts and incorporate it in their own tissue using like specialized cells. They use this to also perform photosynthesis, same way as plants do, and they derive energy from the sunlight. And some researchers suggest that they do this to increase their reproductive fitness rather than like growing or having bigger antennas or whatnot. So one thing that I wanted to look at specifically within these species that intrigued me was their predation tactics. I looked into sea slugs overall and noticed that some of them camouflage into their substrates, while some perform or do aposomatic colorations, and I'll show photos of this in the next slide, and some even have the ability to, like in what I said before, they steal like functional chloroplasts and they use that to put in their tissues. Some do the same thing, but they do it for like toxic chemicals that are found in things like sponges. So I thought that was really cool. I wanted me to look into it a little bit more. Alright, so these are 3 examples of nudibranchs. The one on the left, no, right, you're right, is the one that I took a photo of. These 2, I wish I did, but I did not. And the first one, for me, this is an example of camouflaging, possibly because they ate something in their diet that made them green, and then therefore they're able to steal that functional chloroplast and pigment to turn their own tissues green. In the middle one, that's definitely not the case. This could be still like pigments that they stole from certain food types, but this is displaying aposomatic coloration, basically telling other predators, hey, don't eat me, I'm potentially poisonous. And some sea slugs do present like mimicry, so that's also really cool. And then the one all the way on your right, the lechuga, or Elysia diomedia, that one I wasn't sure about because they had these little filaments at the top that had a lot of different colors, sometimes they were more contrasting, and then their inner like tissues that are on the side that you can't really see in this photo, that always had different colorations. I've never seen like one that had the same one that I was taking photos of. So this led me to my overall question, thinking about how they react to predators and how they have different colorations, of how does perceived threat by potential predators affect the coloration of Elysia diomedia. Alright, so this is collecting data. This isn't me, this is my lovely helper Grace, who is holding a lechuga. They're very, very small. So for my independent project, I did go to Kahini Kill, and that's where I found and located a lot of my subjects. So for my methods and materials, I required only 2 things. I required an underwater camera and then a really, really bright rock, because I kept losing track of the lechugas, or I would try and swim and I'd flap them away, so that was really sad. But stepwise, I had to locate the Elysia diomedia, take before photos of them to see what their coloration was before handling, taking photos of them in my hand when I was handling to kind of simulate a sort of predation experience happening, and then putting them on a different substrate, taking an immediate photo to see if there's an immediate change in their coloration in response to the predation, and then taking a photo 30 minutes after. And after that, there's image J analysis, and then I will get into image J later. Alright, so these are my nudes. This is an example. This is an example of like the 4 different like timestamps of the photos that I took for one of the lechugas. So on the top left is going to be the before photo, Right is going to be the handling photo. Bottom left is going to be when I put it on a different substrate. You see that the substrate underneath is more pink. And then this is 30 minutes after on the right. And then short story, this is my favorite nudibranch that I took photos of, specifically because I had to battle a fish for it because I was trying to eat it. So this is an actual example of the color that it changes to when there's an actual predation attempt, since a fish was trying to eat it. Alright, so this is getting into like tactics of using image J. It wasn't very fun. I had to go through over 3,000 photos, renaming them, opening them, editing them, so that was great. And then selecting the C-slug, I had to go in and select the inner tissues, and then from that, ImageJ provided me with 2 graphs. There was an intensity graph showing me the lightness and darkness values of the nudibranch, and then the RBG graphs, and then the RBG is the red, blue, green, so primary color analysis, telling me which color was more dominant, and that was the most important one that I used. Alright, so this isn't my research, this is what I wished my research and data looked like. It does not look like this, instead it looks like this. So just pretend that this is what it looks like, and then we'll just go with that. So this is my intensity graph for the 4 examples that I looked at. Overall, the intensity in the middle portion, where I'm holding it, which is the second portion on the x-axis, and then the third point on the x-axis being the different substrate, that didn't have as much of a correlation in the middle, but looking from beginning to end of every single nudibranch, they all seemed to decrease their intensity, meaning that they all became darker in color from beginning to end, whether the middle was different or not. So I thought that that was really cool, and that could potentially be due to different types of stress signals, or different reactions to predation, or if they had a certain amount of pigment store that they were able to use or not. This is the color contrast that I use for nudibranch G, F, J, and I, same for individuals. Overall, the contrast seemed fairly consistent. The only one that had a significant contrast decrease would be nudibranch I at the very end. And what I consider a significant decrease is less than 50% of the color contrast being different. So this means that it had the highest values of darkness and then the highest values of lightness at the same time. And that would be symbolized with aposematic coloration, since that's kind of what it is, contrasting different vibrant colors. Lastly, this is the most important one, the RGB values, so the red, blue, and green. This one, it didn't seem as cohesive when I was first looking at it, but when analyzing the graph on ImageJ, the column between 0 and 85 is going to be where the nudibranchs had a dominancy of red. 86 to 170 is where they had a dominance of green, and 171 to 225, which nothing reached above, like 125, was blue. And I thought that was really interesting, since none of them showed a dominance of blue, and they all remained within like a certain column between red and green. So what does this mean? Okay, so back to what I was saying before in the beginning, a lot of researchers have suggested that the coloration of nudibranchs is correlated to the type of algae and the pigments in the algae that they consume. So there's 3 broad, There's 3 broad colors of pigment that occur in algae. So there's green, which is the bottom one, which is going to be chlorophyll. There's red, orange, yellow, and brown, which is going to be the carotenoids in algae, and that's going to be in the top one. And the one that's the most rare and least seen is going to be iridescent green, or red or blue, and that's in the physobilins. So I think this kind of makes sense, just because the green was the most dominant kind of algae that was in the waters that we were looking at, and then the red one was the second most prominent, and then the blue was the least, which is definitely correlating to the RGB graphs that we were looking at before, where blue was just non-existent, and then green and red was more dominant within all of them. So, to conclude, okay, so close. Did I answer my question? Kind of. We do know that there is a certain level level of changes in coloration, whether there is, oh my god, thank you so much. Wait, I think I should get a pausing time. Okay. I did somewhat answer my question. I didn't see an overall distinct correlation with certain colors versus different kind of handling strategies or when I set it down on a different substrate, but I did see that there was a certain range that they stayed in between, potentially relating to the types of algae and diet that they have. Also, lechugas are just really cool, and they're very pretty, and they're really under-researched, so I think it'd be cool to learn about more of the functionality of how they work. A larger population is definitely needed, and a more standardized methodology is required in order to get a better sort of correlation to figure out why they do what they do. I couldn't get actual photos of the instructors, so you guys get symbols, so thank you. Thank you very much.
We have time for one question. Yes, Lisa.
I hope this isn't a myth, but it's my understanding that flamingos have a pink coloration because of something in the shrimp that they eat. Is that a
It's the
similar
pigment in the
mechanism?
shrimp. Yeah, it is a similar thing. The only difference is that if like a shrimp has, like flamingos wouldn't be able to obtain like a toxic chemical from an animal like sea slugs can. Like they solely retain the like coloration. So if flamingos weren't eating shrimp, then they wouldn't be pink, they'd be white. So that's why in zoos they're like always bright pink, because they're constantly only feeding them like the brightest shrimp that they can find because it's more attractive for people to look at.
Thank you. Our next speaker is Vivian Wallace, and she will be telling us about octopuses.
Okay, so hi guys, my name is Vivian Wallace, and I'm a junior at UC Berkeley, and today I will be discussing my research on wild common octopuses and how they can become conditioned to light and food. So introduction, my goal was to obviously condition the octopus with light and food, so I'll go through what conditioning means. So conditioning is the pairing of a conditioned stimulus with an unconditioned stimulus. So for the conditioned stimulus is usually a neutral stimulus. So in this case, it was light that I shone with a dive light. The unconditioned stimulus is biologically potent, like food. So I used bait. And then the unconditioned stimulus is a unlearned reflex to the the unconditioned response is an unlearned reflex response to the unconditioned stimulus. So it would be like coming out of the den to eat the bait. And the conditioned response is a response to the conditioned stimulus when it's presented alone. So whatever happens when I only shine a light. So my central question was, are Octopus vulgaris affected by the consistent pairing of conditioned stimulus, the light, and the unconditioned stimulus, the bait? And can this produce a conditioned response? So from May 13th to May 17th, I studied 14 different octopuses, and I performed 3 trials on each octopus. And so now I will discuss trials 1 and 2, which is different from trials 3. Trials 1 and 2 are both the conditioned stimulus and the unconditioned stimulus. So first off, I would set up a GoPro outside of the den, and then I would go down and I would shine a flashlight into the den for about 5 seconds, and then I would place the bait right outside of the entrance of the den, and then I would leave the area for 5 minutes, come back, retrieve the bait of the weight that the bait was on, and then I'd retie another piece of bait to it, and then I'd do the whole thing again for trial 2. So shine the light, place the bait, wait 5 minutes, retrieve the weight. So the GoPro was running the whole time. So for trial 3, I just shone the dive light, as you'll see. This is the conditioned stimulus, and I waited 5 minutes, and then I turned off the GoPro. So there are 4 different behaviors presented by Octopus vulgaris. The first one usually to be presented was the octopus's body leaving the entrance. you can see right here that that's the octopus's body. It would peek its head out of the den. Oh no. Then it would usually explore the area with its tentacle, as you can see right here. And its tentacle is used for sensory reception, so that makes sense. The octopus then would make contact with the bait, and then finally, it would sit on the bait, eating the bait. It sit on the weight, eating the bait. So first, I calculated the average response times for each behavior presented. So you can see that between trials 1 and trials 2, which had both the conditioned and unconditioned stimulus, they were pretty similar, but in trials 2, the second one, it was a little bit quicker. So this just kind of shows that, yeah, trial 2 was quicker. I did exclude 2 octopuses from this data because they showed extremely long response times due to eel activity. So octopus's biggest predator are the eels, so moray eels. And so you can see one right here, and you can also see one right here. And so in this case, the octopus completely fled the den because he was so scared of the eel, and so I didn't get like good data for 2 of them that had eels. So then I also plotted the time of trial 2 against the time of trial 1, like the response times. And for this one, I did for the body exiting the den, but this was the trend for all of the behaviors that I described before. So in group 1, you can see that the time in trial 2 was quicker than the trials in trial 1, whereas in group 2, you can see that the time in trial 1 was slightly quicker than trial 2. But you can see that there was a bigger difference between the trial response times in group 1 for when the second trial was quicker than in group 2 for when the first trial was quicker. Okay, so now on to conditioning. So 6 of the octopuses showed signs of conditioning, and 8 of them didn't. So the signs of conditioning for the third trial, because there was no bait, would only be exiting the den with its body and also exploring the area with its tentacles. So that was cool. So now I will show you an example of conditioning. So this is during trial 2, when there was both the conditioned and unconditioned stimulus. It's just sitting on top of the weight, eating. And then this was at the beginning of trial 3, while I was shining a light into the den. So you can see it's hidden in its den, doesn't want to come out, it's very scared from the human activity and the light. And then this is an example of what happens right after I shine the light during the third trial without any bait. So you can see the octopus is right here, and it extends 2 tentacles out, feeling around the area where the bait was, expecting the bait to still be there because of the light stimulus. So that is an example of conditioning. So now on to discussion. So these octopuses showed signs of conditioned learning because trial 2, with the conditioned and unconditioned stimulus of light and the bait, was faster on average than trial 1. So we can see that there was an association formed during trial 1 that made the response time in trial 2 quicker. So this shows that they are able to cognitively learn and remember, which basically shows how smart they are. And then, so for conditioning, 6 of them were conditioned, 8 of them weren't, and the 8 that weren't, it could be due to just simply not being conditioned, or it could be due to not having an appetite to go out and get more food because they've already eaten 2 pieces of bait from trials 1 and 2. And additionally, it could also be because they are just very scared of predation. So they don't want to leave that den again after leaving it 2 times. And also, so finally, I would say, okay, well, basically, I didn't do anything. But this is good, I can look at your finger. Okay, this is good.
to adjust to their environments and helps them interact with important resources and potential threats. Whether this is outcompeting organisms for food or learning to recognize a sign of danger, the octopus vulgaris' ability to be conditioned may have driven their evolutionary success and is a testament to their intelligence. So just like Pavlov and his dog, I created Wallace's octopus. Biologica and my Quantico Peers. And finally, is my favorite video, showing how cute they are and how curious they are. Hi. Yay.
We have time for questions. Ben?
Yeah.
Ben?
What did you use as bait?
I used this type of sardine that we got from the fisherman in Quantico called Guyera sardines. I think something like that. At least that's what the miner told me. So yeah.
How much time passed between each trial? Like, was it days or hours or like?
It was 5 minutes.
Okay.
Yes. So I could do a lot in one day.
Okay.
If you had more time, what else would you do?
I would test whether the association that I expected to be from the light was actually from human activity. So I'd take out the diving light and I would just the third trial would just be me diving down and looking at it and leaving to see whether that was the association instead.
Do you expect following these results that you see almost like a negative conditioning, like if you were to do something they associated to be negative, that they would do something in response, versus this is like a positive stimulus?
Yeah, I feel like it could go either way. I'm not sure what hiding in the den, if it was outside of the den. Oh, definitely. I think they do that anyway. I think they already have a sense of risk and that's kind of already being conditioned into them.
Thank you. From UC Santa Barbara will also be telling us about her studies of octopus behavior.
Yes! My name is Alicia Benedict-Moran, and I do go to the University of California, Santa Barbara. As Vivian just said, octopuses are super cute and super curious, and that's why I studied the octopus vulgaris behavior when presented with novel items. Are they neophilic or are they neophobic? First, just in case you don't know, neophilia is the wanting or loving of new items, and neophobia is the opposite. It's the fear or dislike of new items. So now, octopuses. They are extraordinary alien-like creatures. They have 8 appendages equipped with 1,000 little suckers that also has minions. This is going to be really good because now you know so much because we just talked so much about octopuses. But they have thousands of little suckers that help with sensory, smelling and tasting, all things like that. And they're known to be amongst the most intelligent creatures. So I don't know what contributes to them being intelligent. Can it be their anatomy, so just things like that they have the advantage of having 8 appendages? Can it be their willingness to explore and wanting to learn? Could it just be their environment? Could it be how we were in a fishing town in Keel and they're being hunted every day and so their goal is to hide, or just could it be anything else about their environment? It could be their curiosity. So my central questions were, do octopuses display a similar level of willingness to explore a puzzle when presented with a food reward? And to what extent do octopus behaviors differ among individuals? Essentially a really in-depth analysis of their behaviors. Okay, my materials and methods. So what I did for my experiment was I have 2 sets of trials. And the first set of trial is
3 glass bottles. They have sand, and one of which has a piece of bait, which is the same sardine that, sorry, not octopus. Vivian used in her octopus experiment as well. We can see it, like the middle bottle right here. That's the piece with sardine. And this is just a good video because it's like really easy to see just like what I did and what happened. Okay, and then, so I put them down. Well, first, Stephen would go find the octopus. I put the camera down about like a foot and a half away from the den, and then I put the 3 bottles down. I press record, and I'd leave. After 10 minutes, I'd come back, pick up the items, replace them with exactly this format, but 3 PVC pipes, also stuffed one with bait, and then blocked on both sides with rocks. And the other 2 were just filled with rocks, there was no bait. So the goal was for them to pick the right item to explore, and then get the bait, and then for them to just explore what they want to do, and me analyze the behavior. Okay, so then put the 3 pipes down, leave. Wait 10 minutes, come back down, get the 3 pipes, get the camera, get everything, say thank you to the octopus, and leave. Video. This is Octopus Felipe. You'll learn that they all have names. And he comes, and he's funny. You'll see he explores, he puts his tentacle in that one on the left. Bam. Done. Comes over to the middle one with bait. You'll see the bait move. Maybe you won't. Did you see the bait move? Okay, that's all you need to know. He touched the bait and then decided to leave it. He moved the bait with his tentacle, and decided to leave it. And why did he do that? I'll show you in the results, but he was the only one that touched the bait and then just left it there. Like, I don't know why he did that, but that's just his personality, and that's okay. Glad you giggled. All right, headshots. We got Gutowski, Felipe, Heidi Klum, Kickflip, Novio, and Porcupine. Novio, yeah, I'm just going to answer that question really quick. Novio, it was a pair of 2 octopuses in this den, so originally they were named Novios, but only one came out, so Novio was just one. All right. So this is pretty much all of their action shots, and the only one that doesn't have like an actual headshot is Porcupine. And that's because he didn't want to come out, and that's also okay. Alright, so the main points of my analysis were to observe their observational behaviors, their exploratory behaviors, their defense behaviors, and their latency in coming out of their den, and whether or not they captured the bait. This part's going to get lots of data, alright? So we have, of the 6 octopuses that we have for both of these trials, only 1 of 6 for the 3 glass bottles test was successful. That means I have 5 minutes left. So was successful in capturing the bait. And this was Gutowski, who's really good. But I highlighted Felipe, who was the video that I just showed you, who spent 80% of his 10-minute trial time interacting. This means like interacting and touching the actual item. So 86% of his trial time interacting with it, but not retrieving the bait. And like why? Maybe he wasn't hungry. Maybe he wasn't into it. I don't know. He's fine. I mean, he could be different. Okay. And then we have the 3 PVC pipe tests, of which 3 were successful in retrieving the bait. Not Gutowski. Although Gutowski did spend 41%, he's the first column here, although he did spend 41% of his time interacting with the second item, he did not successfully retrieve the bait. But Heidi Klum, who did not in the last trial here, did not in the last trial come out at all. Kickflip also did not come out at all, spent so much more time interacting with the item. And then Porcupine spent 61% of that. So as you can see, and this is like a pretty probably easy trend to see, the more time they spent interacting with the item, normally they got the bait. It was just Gutowski that didn't, and then Felipe that didn't in the last one. Bam, done. Alright. This is a comparison graph. Pretty easy. 3 glass bottles, the trials are green. Gray PVC pipes, the trials are gray. And so what you really want to be paying attention to here is the split up of the 3 boxes. So the first box that we have on the left is the observational behaviors, which are labeled as quick peeking out of the den, log observations that are more than 10 minutes, whether or not they were perched in front of the den at all. And then we have the middle section, which is observational, which is they reach out for the item, they move closer to the item, they actually touch and explore the item, and then they have their tentacle inside the correct item. And then we have,
inside, which only one put their, or a couple put their head inside, but not here. Okay, and then the last two, in and out of the den, and then dirt plume, were defense behaviors, which is like when they come out and then come back in, they're a little bit scared, or if they throw out like a dirt plume. Kind of hard to describe, but they throw out a dirt plume when they're deciding whether or not something is a danger to them. Okay, this is a latency table. So this was between the two trials, how long it took for each octopus to come out of their den. And for Felipe, Heidi Klum, Kickflip, Novia, or Kickflip, and Porcupine, all of the negative numbers mean that they took way less time coming out in the second trial. Although Kutowski, weird, spent way more time coming out in the second trial. But I think that Kutowski did this because he got the food in the first trial, and maybe, yeah, could have been full, could not wanted, although he like saw the bait, explored the item, and was like, oh, I don't need any more, so like fine. But then, I'll hurry up, sorry. Okay? Whatever. The octopuses took way less time coming out in the second trial, so that just shows that they're like quick learners, and that could also be because they're like, oh no, well now they took away my food source after 10 minutes, like I gotta go really quick. Or now I'm not scared of these like stationary items that I've already tested and observed for a long amount of time. I can come out and interact with it and I'll be okay. You know? Alright, results and discussion. Essential answers to my questions: do octopuses display a similar level of willingness to explore, puzzle, and present to a food item? No. To what extent does octopus behavior differ among individuals? It differs a lot. All the octopuses I've studied behave differently, and the difference in their personalities is really easy to observe. And here's a really simple graph that I'll explain to you super quick. So 0 to 40 is their behavior score. 0 is timid, shy, not really wanting to come out. 40 is rebellious, rambunctious, rowdy, wanting to come out of the den. And so at the bottom we have Novios and Porcupine, scores 7 and 8. At the top we have Kutowski and Kickflip at 28 and 36. And these scores were evaluated on just the ranking that I gave those 11 behaviors that I gave to you in the previous graphs. I gave them scores from like 1 to 5, and then ranked like how extroverted or essentially like introverted, euphilic, or euphobic these octopuses were to coming out and interacting with items. Okay, the adaptive significance of curiosity. What I really fast. You can kind of associate this to like children. And so what children do when they're learning things for the first time, they are observing, and then they're exploring, and then they're touching, and then they're kind of going for that. So with that curiosity, they're building experience. And with that knowledge, or with that experience, they're building knowledge. And knowledge equates to like a higher level of intelligence. Is that right? That was basically it. The importance of studying these organisms is that they have really amazing and cool behaviors. And if I were to do this again, I would definitely try to do it again with another set of items, like another set of 3 items, like 3 PVC pipes, 3 plastic bottles, maybe like 3 jars or 3 anything else that would give them more time to like be acclimated, and we can prove if they're just getting more comfortable with these novel items, or if they're just getting more comfortable with these novel items, or if there's something that goes on behind the behaviors. My conclusions is that octopuses are curious, intelligent, and lovable animals. They're very uniquely behaved to each other, and I think they deserve roles in society.
Time for one question.
Yes.
Emma.
I thought it was interesting that the Novios had like the lowest behavior score, like because you said that meant they're like less likely to come out of the den. Do you like notice when octopuses are like interacting with each other? Does that like did any of the behavior traits come out? Because those are the only 2 that were like in a pair in the den, right?
Yeah.
Like
So
how did you see them interacting with each other and like how
they actually are like really antisocial creatures, so they don't interact with each other essentially at all. So it was really weird to see both of them in the den. I do think that we saw, it was essentially like a big coral bed, it's the Novios den, and I saw 1 coming out and I saw 1 in. So I know there was 2 in there, and I was like, well, this is cool, like what can happen? But at the end, I like kind of put my head in it and like looked around, and I only saw the 1, so that's why I didn't
other because they're really abnormally together. Like they're never really together.
Thank you.
Wait, do you want me to put my computer on?
Say it again?
Do you want me to put my computer on it? Or I did email it to you like probably an hour ago.
An hour ago. Okay, let me see if I can. Do you have a Mac?
No. I think my computer can take the new like little Mac thing. That's what I'm
All right, let's give it a try.
Yeah.
Do you have ECDC? Do you have ECD port?
I'm not good with the terminology.
Yeah.
Try it on. Put it up here. Is it on? Okay.
The next speaker is Lance Woods from UC Berkeley, and he's going to be talking about brittle stars. Wait, is this part of the presentation?
No, I just want to go. Kind of like do like a TED Talk walk during the same time.
Oh, okay.
It's a power guys. Oh, look at this guy too. Okay. Oh, that's gonna be fun. Okay. Brittle stars. Enough said. Questions?
What are brittle stars?
We're still trying to figure that out. I cut all my slides. Easy.
What was your project?
When you like it now. How much time do I have to go?
You got about 8 minutes.
Yeah. Questions?
What did you do?
I studied brittle stars.
How?
I found
And what did you record?
what you want to get in the nitty gritty of it. I'll casually do it. Alright. Brittle star characteristics. Oh god, man. I gotta work on my themes. They're pretty boring stuff. Gurika, Costa Rica, Lance. This is a new thing I'm trying. I'm trying to use my middle name in my name when I speak. So, Lance Tyler Olch. Okay. These are brittle stars. I probably don't even point my finger and do this at the same time. But, boom. They're pretty cool. I love them. They're unbelievably beautiful and kind of alien looking. Kind of like the Alien movie franchise a little bit. If you've seen those, you should if you haven't. They are echinodermatas, which is like a family or order of sea animals. Sea stars are in there, sea urchins, invertebrates of the marine kind. If you look at them, what's one thing you notice? No eyes, which is crazy. But they do have light receptors in the little arms, these little side arm things. They can perceive where light and dark are, which is pretty cool. You know, I don't even know how. Someone probably does. They live under rocks, and how they feed is they put their little arms up under rocks, and they collect little debris as it floats by, which is pretty cool. Pretty good diet. They reproduce sexually and asexually. So sometimes they can like cut them, like they can fission or something like that. They cut themselves in half and then the two grow into whole other brittle stars. And then they also bone at the same time. So that's cool. Well, we'll get into that for my discussion later. They move, the cool thing about brittle stars, which is, maybe like, oh, why would you say sea stars? They are different from sea stars. They move very quickly, and they wiggle their arms to move. In a demonstration, say like my 5 limbs are like a limb of a brittle star. They have one, say it's my head, that points them in the direction they want to go. And then they kind of like, as if I'm like an inflatable dude at a car sale. They push themselves, like, see, this is the ground, I'm pushing myself forward. And then say like, my eyes are closed, I just feel the light, and the light is now over here. This arm becomes an arm that doesn't move, and then like, and then you stop, and then like, it'd be like a stanky leg as if they're going this way. So, I do have videos, but I just couldn't figure out the technology for that. So, that's related back. Research questions, because Frank made me get one. I just wanted to look at them, really. What are the chances a rock has a brittle star under it? And how long, slash is the sign, far does it take them to resettle after removed from a rock? They really much do not enjoy being in the open, which is a bummer, because then I can only spend a fleeting amount of time with them. But hypothesis. For my prelims, which is short for preliminary observations, which is a term we use in science, as a majority of rocks will have brittle stars for my prelims. And predominantly, I'll do probably a big note to really hit on. There's 2 species I found for the most part. Black spinies, Alexander spiny, this one's like gray and white, this one's like all black. This one, missing an arm, it'll grow back. Do not fret. So I think a majority of rocks will have brittle stars, and then those will be black spinies. It will take them about 5 seconds to 5 centimeters to resettle. Let's get into the nitty gritty here. Also, just look how cool they look too. Look at that. Just so alien-esque. It's kind of cool. It's kind of a deep one here. I swim down to a rock. I start recording. I lift the rock, which is trickier than it sounds when you're in the water. Most things are slightly harder. So don't take this for granted. It wasn't very easy. I continue recording. I stop recording. And then a little 5.5. I didn't do this every time. I switched to 5.5. I measure the distance they travel. I stop recording once they stop. And then that's supposed to be 6. I swim back for air. And that's what made the whole thing quite difficult. I could not hold my breath for very long. I do sometimes enjoy a cigarette from now and then. So it was a good test for me. Okay. Abundance. This is just something I was curious in. It wasn't really my questions, but this is the total amount of individuals I found. All my graphs are pretty elementary. My graph game is pretty bad. So this is just, I think I found I should have put the numbers on them for sure, now I'm looking at it. But if I had to guesstimate, it'd probably be, I think I found 94, or 84 of these guys, and 79 of these guys. Of the black spiny, this is the Alexander spiny. Which is quite a lot of dudes, I will say. I saw quite a lot of them. This is the distance they traveled. This is what we call a histogram. Bars, little numbers. This is 0 to 5, 5 to 10, 10 to 15, 15 to 20, 20 to 25, 25 to 30, 30 to 35, 35 to 40, 45, 50, 55, 50, 55, 60, 65, 70, 75, 75, 75, 80, 85, 90, 95, 95. Exactly what I was thinking. Alright, so black spiders, they don't travel within the 5 centimeter very often. This is the number of individuals. But they love that 5 to 10, that 5 to 10 range. That's their track. That would be their sprint for sure. And then it's kind of like a little bit of a roller coaster. We start small. Moving on. Kind of a similar thing going on with the Alexander spiny as well. So that's pretty similar. Definitely going over that. So this is, again, 0 to 5, basically the same amount of individuals. And there was less Alexander spiny distances I got because as time went on in the weeks, I was getting lazier for my project. And I just didn't get that much Alexander spiny data distance-wise. So, but also kind of like a roller coaster again. You start low, like, woo. So yeah, they really don't like instantly find. They take. I feel like to me this represents they take a little bit of time to find. Well, they go a little bit distance to find a good hiding plot, not just the closest one to them. And also some of them go fucking far, man. It was 95 centimeters for like a little dude that doesn't like to be out in the open very much. That's pretty far. So good for him. For the chances of rocks having brittle stars, which is a question I asked. 84. So let's piece this guy together. It's a little confusing. Blue is alone. So that means one brittle star. Together, there's like multiple brittle stars under a rock. So all these rocks have brittle stars. This one is a 0 second, no brittle stars. 0. And then sometimes I also do this just for the kick of it. Multi-species rocks. So that means like there's one of those, both under a rock. Didn't happen very much, but did happen. So based on this, I crunched the numbers. It took me a while. Essentially, 84 is 50% of all that together. So about 50% of rocks have brittle stars, which is cool. Pretty perfect number. This is the resettlements at the time. It took them like I took away a rock, they were under a rock. Go run away, go into a new rock, stop the timer. That's this time. Again, black spinies. They're not in that 0 to 5 second range. They're definitely more than 5. Even like, I'll push it to 30, like a pretty solid chunk of them here. Mostly the 5 to 15. They take the time. Some of them, again, take a pretty goddamn long time. And those were unbelievably difficult to record because I cannot hold my breath again for very long. So then this is the Alexander spiny resettlement. A lot more in the 0 to 5 range. And also just like, a lot more, like, not that many, 25 to 30. They're a little more quicker. This one's a little more out. So again, I think the black spines, they're a little bit bigger than Alexander spines. I think they're more comfortable taking the time and going a little further distance than Alexander spines were. In conclusion, look how cool these guys are too. These are some weird looking ones I found. Brittle stars are very cute. I am in love with them. They're amazing. The black spiny brittle stars take slightly longer than Alexander spiny brittle stars. I would say these guys are in the realm of like average 10 seconds, 10 to 15. These guys on average, probably more the 5 to 10 range. So I was definitely wrong with the 5-second claim in my hypothesis. I definitely kind of came up with that hypothesis after I did the experiment. It's not really a true hypothesis in my opinion. But I want to make it sound good. They range in distance from 1 centimeter to 95 centimeters. On average, Alexander spiny brittle stars take shorter distances to find new settlements. And just about 50% of rocks have brittle stars. So go out and look for them. They're pretty cool. These are my acknowledgements. Dude, this is Frank. He wanted to take a photo with me on this island. And I was like, for your benefit, I won't smile because I know you don't like to smile. And he said we should. So look at that. Yeah, I'm pretty proud of that one. This is a poem Katie wrote about Casa Verde. I guess like I didn't really have photos of like everyone, just like thanking everyone that was there in Casa Verde and all that place. This is Alicia. This is where we were doing all our experiments. And yeah, thanks everyone.
Unfortunately, we don't have time for questions. However, the speaker will be in the refreshment area after our next speakers. Ooh, thank you.
That was so lit. Were you like joking? Were you like say the first song?
Our next speaker is Grace Spencer from UC San Diego, and she will be talking about microplastics in crabs.
Okay, so hi everybody, my name is Grace, and this is my research presentation, Feeling Crabby, Microplastic Prevalence in Grapsidae Crabs of Queensland. So to start off, I'll provide some background on microplastics. So since the 50s, plastics have been growing in popularity and demand, and now about 350 million tons of plastic waste are produced every year. And of this amount, about 75% eventually ends up in the environment, mostly in landfills, rivers, and oceans. And the problem with this arises from the fact that microorganisms can't degrade plastics like they do for most materials due to their chemical composition. And therefore, plastics are only broken down by UV rays and weathering into smaller and smaller pieces, but they maintain their same structure. And when they become smaller than 5 millimeters in length, they're categorized as microplastics, and microplastics are very easily consumed by organisms in the environment because of their small size. And this presents health consequences for organisms like internal tissue damage and blocking digestion. And then you can see here some of the plastic pollution found in the mangroves of Tahiti Keel. So rivers with plastic waste sometimes flow through mangrove forest environments before reaching the ocean. And because mangrove forests act as a kind of filter between the river and ocean, they accumulate a lot of plastic debris. And this presents a significant problem for the animals that live inside them, including crabs. Crabs are especially sensitive to microplastic contamination, and some species that even be recommended as bioindicators for microplastics. In my study, I chose to look at Grapsidae crabs because they're omnivorous scavengers and they're present in intertidal zone habitats of Tahiti Keel. And I wanted to investigate whether mangrove crabs, like crabs present in the mangrove habitats, consume more microplastic than those outside the mangrove habitats because mangroves are filters, which led me to my central question: does microplastic prevalence vary among Grapsidae crabs based on mangrove forest versus offshore island habitat? And to address this, I wanted to do it in a way that minimized harm to the crabs. So I collected and analyzed feces by looking at particle counts under the microscope. And these are the two habitats I looked at. These are my two sample locations. My study was conducted in Guanacaste, Costa Rica, near Tahiti Keel. The first site here was in the mangrove forest along Rio Tahiti Keel, about 300 meters inland of Tahiti Keel Bay. And the second site was Bajo Rojo, which is a rocky, offshore island about 4 kilometers off the mangrove forest. These are the two Grapsidae crab species that I studied. On the left, we have the one present in the mangrove forest. It's called Gonapsis pulchra, or the racer mangrove crab. And on the right is the one present on Bajo Rojo, which is Grapsis grapsis, or the red rock crab. So for an overview of my method and materials, first I would go to either location. I would capture several crabs. I would measure them, and I'd record their location and size for future reference. Then I would hold them in captivity for several hours until they defecated, which usually took around 5 hours. After this, I would collect their feces, take the crabs back to their habitat, and then analyze the samples. I did this by splitting each feces collection into 5 subsamples, and then I would count the number of microplastic particles under the microscope. To identify the microplastics, I looked for consistent color and density, a lack of pattern, a lack of biological shape and structure, and also bright color and especially long length for microfibers. And then in total, I was able to collect 105 subsamples from 13 crabs. And also, since feces collections had small amounts of water, I took 60 water control samples, 30 per site. So with all of this, what did I find? First, 93% of the studied crabs had microplastics in their feces, so they showed evidence of microplastic consumption. So every crab except for one from Bajo Rojo. And then now moving into comparing between the two sites. On the x-axis we have the different categories of subsamples with mangroves in green and Bajo Rojo in red. And on the y-axis we have the average number of microplastics per subsample. So you can see that on average mangrove subsamples had higher abundances of microplastics within their feces. However, when I analyzed these differences with the t-test, the difference was not statistically significant. Also going back to crab size, this is a scatter plot showing the relationship between crab size and microplastic prevalence with carapace width on the x-axis. If you don't know what that is, it's like the shell on the main part of the body of the crab, so like excluding the legs. On the y-axis is the number of microplastics in the fecal subsample. And each subsample is plotted as an individual point, and the repeated points are more dense. And then I analyzed this with lines of best fit and find the R-squared values. And based on the lines of best fit being very flat and the R-squared values being very low, I concluded there was no correlation between crab size and microplastic prevalence. These are example images of some of the samples that I looked at. So
on the left we have one from the mangrove forest. It's a little bit darker and more dense. And on the right we have one from Bajo Rojo. So what does all of this mean? Primarily, it means that microplastic contamination and consumption by crabs is a very ubiquitous issue. The fact that microplastics were common in grab-studded crabs across all of the habitats and sizes indicates that there are very large amounts, especially because when I was doing my research in the mangrove forest, I saw lots of large plastic contamination pollution, but on Bajo Rojo I didn't see any. However, since there were microplastics in both environments, it indicates that microplastics are so present. Sorry, I lost my train of thought. Microplastics are so prevalent, they are present even without local macroplastic pollution. This presents a problem because it's harder to focus treatment or prevention efforts by location. For instance, if I had found evidence of more microplastics in the mangroves, then I could have made an argument for focusing cleanup efforts along the Coconino Keel, but since this wasn't the case, it was harder to target the issue. These microplastics likely came from populated areas both near and far. So some could have come from the local village of Coconino Keel, but others probably came from faraway places and eventually reached these crabs through traveling through the water. And this plastic pollution came from mismanaged disposal of plastic waste, like overflow from landfills, and plastic that never made it into landfill littering. And I also want to note that most of the microplastics that I found were in the shape of microfibers, which indicates that objects like clothing and rope are significant contributors to microplastics consumed by crabs. And this matters for crabs because it has negative biological impacts. Research has shown that microplastic consumption can lead to decreased feeding and growth scope. Also, microplastics in gastrointestinal systems can translate to brains and other organs like ovaries and testes. Research on fish and copepods indicated a potential this to cause behavioral disorders and decrease reproductive success, which endangers both individuals and entire crab populations. Also, this can impact humans and other organisms because crabs are low trophic level and are eaten by things like fish, turtles, and people. They're a very popular seafood. Over 400,000 tons are imported per year. And so this presents a danger to humans because microplastic consumption has been linked to increased oxidative stress, organ inflammation, and cancer risk. So in conclusion, microplastic contamination and consumption by crabs is a very widespread issue. Microplastics are highly present in intertidal environments like mangroves and small islands, and they are consumed by organisms within them, such as crabs, causing biological harm. This is ultimately the result of high plastic usage and waste by people, and so there's a high need for reduction in plastic demand through more responsible consumerism, cleanup efforts, and more responsible management of waste, like with recycling and properly containing landfills. And I want to thank my primary advisors, Frank and Emmy, for providing lots of feedback for me. Also, Mason, Antonia, and Stephen and Minor Junior Laura for helping with my fieldwork. And my hometown floor, Monteverde Institute, Exactamente Logica, and UCEAP for providing an excellent environment for conducting research.
That's time for questions.
What was your favorite crab? I would say probably the mangrove one. They were a lot smaller, but they were also they ran around a lot more, and they also would pitch you a lot harder. Nice.
What do you think was the most sassiest crab?
Probably also that one.
I guess kind of on the same thread. What was your most difficult capture of crabs?
I would say the mangrove ones because, well, the island ones, they were pretty easy to catch because there were a lot of them, and I went with Steven who just grabbed them with his hands. But the mangrove ones, they were much smaller, and they would run and hide in fiddler crab burrows, and we caught them by cornering them in a burrow and putting a container over the entrance, and then we would shove them in.
I saw in one of your graphs you had a control. Can you talk a little bit more about what your control was?
Yeah, so basically when I collected the feces, they would have amounts of water also in the site. So I wanted to assess if some of the microplastics could have come from the water column instead of being in the feces. So that's why I took control samples for the water. And I concluded that it didn't really have that much of an impact because there was a pretty significant difference between microplastics in the water and feces.
Any more questions? I have one. You hinted at this, saying that you didn't know where the source of the plastic was. If you could do one thing to address this problem, one thing, what would
collection on plastic flow from the Rio Grande Keel out to the bay.
Morgan. Thank you. Sure. Our next speaker is Elise Garza, and she will be talking about microplastics in coral.
Just to make sure I got this down. Okay, cool. So I'm going to be talking about microplastic presence across the bleaching gradient in Posolopora coral. And as you guys know, my name is Elise Marisol Garza. So to conduct this research, I collected coral fragments, which we can see right here. This is a very healthy coral fragment that I found in Bahia Hachal, and my whole study was conducted in the Bahia Guajillo. So Posolopora coral and coral overall play a very important ecosystem role. They provide habitat for millions of species worldwide and provide breeding grounds, eating grounds, as well as places for organisms to hide and to live. So this makes them incredibly important for biodiversity preservation. They're nidarians, and they're also closely related to jellies, not coral, a little redundant, but you can see in their polyps that they have kind of tentacle-like structures. Yeah, like right here. In fact, some people, when I've shown them photos of polyps, ask if those are anemones because they're very similarly related. Coral are colonial organisms, and a singular coral animal is a sole polyp. So it's one of these right here. So the goal of my study was to measure microplastics within the digestive system of polyps, so especially in areas such as the gastrodermis. Understanding coral and understanding my study, it's critical to know about corals' zooxanthellae symbiosis. Corals are dependent on this intercellular symbiosis with zooxanthellae, which provides them with a critical nutrient exchange that keeps both of the organisms alive. The mutualism is extremely important for coral health, but it's also extremely vulnerable, with many environmental changes resulting in degradation of this mutualism, such as water temperature change and pollution, and in some cases, microplastics. So the breakdown of this mutualism causes very high stress on the organisms, color loss, and can make the corals prone to death. And it's called coral bleaching. And we can see some bleached coral in this photo right here. So microplastics are one of the grave threats that corals are presented with. And microplastics, as Grace just covered, are plastics that are under 5 millimeters, and they're formed with from the weathering of larger pieces of plastic. And they come in various forms, such as beads, fibers, fragments, and pellets. And they can have major health effects on organisms, and they're now found in pretty much every ecosystem and trophic level, unfortunately. So microplastics have varying physiological effects across different species and different genera, with coral having a range of effects when exposed to microplastic, including ingestion, rejection, bleaching, tissue necrosis, and lack of sizable negative health effects are measurable. And then cnidarian algal symbionts also have a range of effects, ranging from complete symbiont abandonment to no change in symbiont density or health, meaning little bleaching versus extensive bleaching. Posolopora corals, which is my study genus, have shown consistent patterns of ingestion and negative health effects, such as repression of detoxification and immune systems, among other things. This led me to my research question: is microplastic prevalence correlated with the extent of bleaching in Posolopora coral in the Bahia Guajillo? To answer this question, I followed the following steps. With my materials and methods, I started with identification of coral fragments in the water. I identified coral across 3 different bleaching extents: live, healthy, and partially bleached. and then I would collect them and store them in the water until we left. And then I would transport them in a cool place, such as a cooler, with limited sun and heat exposure. And then I would store them in a temperature-controlled water bath or in the fridge to prevent further coral death. Then upon starting my lab work, I would do 2 controls, a stereoscope examination and a water control, and then I would rinse them with fresh water and grate the coral polyps to get polyp tissue examined. And that's what I would do my primary sampling from, and I would do 3 slides from each fragment. I had 4 study sites, with 2 having the majority of my observations, those 2 being Bahia Tomas and Bahia Hachal, right here and right here. Then I also had fragments from the Muelle and from Bajo Rojo, but collection here was harder. So I had 3 coral bleaching levels, like previously mentioned, which were my live corals, my partially bleached, and my bleached fragments. I had my 2 controls, which were my stereoscope and my water control, which I used to control for microplastics that might be attached to coral externally. And using these controls provided more certainty that microplastics found in my main samples were found within the polyp body and not on the polyp surface, and would indicate ingestion. And in this photo, we can see a microplastic that I found on my stereoscope. To count something as a microplastic, they had to meet 4 out of 5 of these qualifications, with the first one being an immediate strike through. So yeah, essentially, no cellular structures visible, steady shape, small size, steady color, and bright black or clear color, indicating that this is a microplastic and this is not due to the cellular structure. The results were that I had no significant association between bleaching level and microplastic presence, and all of my microplastics found were microfibers. On average, I found 1.28 pieces of microplastic per fragment that I assumed to be internally located based off of my controls. My results showed that there was difference in the ranges and in the means between bleaching levels, with negligible bleaching having less on average due to its mean, partially bleaching having a little bit more, and then extensive bleaching having a higher mean but also a higher range from the previous 2. However, this did not denote association. And we can see the difference in mean here that follows with bleaching level. So, what do these results mean? We can see that microplastic presence occurred frequently in Posilopera coral. I had 12 out of my 20 samples showing fragments showing microplastic presence, and this follows past research in microplastics in Guajimi keel across trophic levels and organisms that previous EAP students have studied, and Grace's name will soon be added to this list. This also follows Posilopera coral microplastic ingestion patterns that have shown consistent microplastic ingestion across the genus. And it also follows patterns of, it also shows that there's no association between symbiont density and microplastic, following Tang's research about Posilopera not changing their symbiont density after short-term microplastic exposure. While my methods were different and I studied more Posilopera than singular species, we came to similar conclusions, indicating an overall genus trend. Additionally, this showed the fact that there was little association between the 2 variables, showed that there was a probable effective rejection across bleaching levels, and if we assume that to be by the same mechanism that sediment rejection is, it would follow Hicks and Long's research of consistently productive sediment rejection functioning across bleaching levels, with no decrease of sediment rejection as coral becomes more bleached. So, to conclude, species and genera-specific physiological effects, such as the ones shown by microplastic in corals, call for more research, as generalizations are not applicable nor responsible in looking at coral health. And though there's no association between Posilopera symbiont density and microplastic presence, it's important to research health effects beyond bleaching on a species-specific basis. Additionally, we conclude that microplastics have deeply infiltrated natural ecosystems, and making it all the more pressing that we actively work against microplastic further penetration of ecosystems and identify sources. And acknowledgements, thank you guys so much to everyone on the slides and everyone in this room.
Let's do one question and then break for refreshments.
Okay.
Questions?
Great. Since you spent so much time collecting coral, did you happen to notice like the bleaching levels in general across the bay? Like how much of the reef would you say was like healthier?
Yeah, I would say that actually a decent amount of the reef was pretty healthy. There was definitely pockets and sections that had experienced pretty intensive bleaching, but I know that there was some more intensive bleaching events that occurred a couple of years ago, and I think that the reef has pretty successfully recovered. Hachal was by far the healthiest site in my opinion, but Bajo Rojo also had pretty healthy reefs. It just was more interspersed and less constant. And I did see some bleaching in Bahia Tomas, and yeah, but I would say overall pretty healthy.
We're going to do one other question.
Sure.
Silvio.
Since you referred to sediment rejection mechanisms, that made me wonder, and I guess a similar question would be relevant to Grace as well. But are the corals then able to recognize some microplastics and avoid ingesting them, do you think? And does that mean that, would that depend on the characteristics of the microplastic? Would they be more likely to ingest some than others?
Yeah, that's a great question. Thank you. So I don't know exactly if they're recognizing when they're rejecting. However, there have been studies that have shown that like across different species, they like use chemoreception to differentiate between microplastics. But in those species of coral, they're actually using chemoreception to actively choose to ingest the microplastics, as they have the microplastics often have like certain characteristics and chemical compounds that make it like beneficial for ingestion, or not beneficial, but like I guess tempting in a chemoreception like sphere, I guess. Yeah, thank you. I definitely need to do more research on that, but that's what I know.
Thank you, Lisa. So we're a little behind, but we're also behind in the refreshment area. So we're going to take a 15-minute break. But before you move, people who are in the next session, please come forward and make sure that your presentation is on the computer.
Can I bring back some more?
No, it's not.
back to the symposium. As you hear, Frank, we're studying. This is the second session, actually the most interesting section of the symposium. And it's about biology of vertebrates. Our first speaker, Jenan Isabel, who's there, from UCEAP Turkey, is going to speak us about project development and the changes in time.
Yes. Hello everyone, I'm Jenan Isabel Osier. I'm a student at UC Berkeley, and today I will be presenting about inert distribution and richness along the tropical montane elevation gradient. And if this was accurate, it would also say Eladio Cruz up here because I could not have done this project without him. Okay, so just for some background information, because I certainly did not know what anura meant before I did this project. Anurans are frogs and toads, and basically in Greek, anura just means tailless. So it's tailless amphibians. And another important aspect of this project that I'll touch on a lot is Holdridge life zones, which is basically a system of distinguishing ecosystems based on precipitation, temperature, humidity, and elevation. Two other key aspects of this are anuran sensitivity. The reason anurans are such strong ecosystem indicators is because of their relative sensitivity to their ecosystems, which means if there are changes in their ecosystems or introductions of harmful things such as pollution, anuran populations are most likely to change right away. So surveying anurans is a great indicator of the state of their ecosystems. And then lastly, this project is a continuation of methods done by both Elizabeth McDonald in 2019 and Taryn Cornell in 2023, exactly a year ago from this program. And so both of their programs were done spring in the same weeks as our survey periods. And so that's why I was able to use them as a reference point for my data that we'll look at later on. And so that comes to my research question. How does anuran species richness and abundance change along an elevational gradient? And how does this compare to the past surveys that I was talking about? So to begin, we can see this beautiful map made by Taryn Cornell last year. And we can see the 4 study sites that all three of us used, which included Santuario Ecológico, which falls in the second of the 2 through 5 Holdridge life zones that I was surveying. Then Rachel and Dwight Crandall Reserve, which falls into the 2nd and 3rd categories, which is right behind the institute. Then Estación Biológica Monteverde, which was the 3rd and 4th. And then lastly, Estación Biológica San Gerardo, which was the 5th zone. And so the way that it worked in each of the sites was that Eladio and I, and also FJ on certain nights, would go out for 1.5 to 2 hours, and then we would spend 3 nights in each site using both visual and auditory survey techniques, which basically meant we would go out, as you can see Eladio here, we would survey leaf litter, and also we would survey the auditory environment by distinguishing male calls. And so we would tally all of this up. And then lastly, this was one of the coolest frogs we saw. This is an emerald glass frog, and that was really, really cool. You can see the little heart right there. That's awesome. Okay, getting into results and discussion. These 3 points are kind of key takeaways that I'll go through before we get into my figures and tables. And so overall, we found 521 individuals over the 21 survey hours. And of these 521 individuals, we found 17 species across 5 families. The species Diasporas diastema, Dualmanogilla rufioculus, and Creogastor precipitatus were observed in relatively high abundance, but mostly were observed in San Gerardo, which we'll see in the next slide. And then one thing that was super interesting is that this survey was more similar to McDonald 2019 than Cornell 2023, which contradicted my initial expectations going into the project. And so this is a pretty big table. It's a little confusing, but basically here are all of the species broken up by family in the green rows. And then each of the subsequent columns are the different sites that we were in. And so as you can see, there was high species richness in San Gerardo and also in Estación Biológica. And then at the bottom here, we can see the number of species as we moved across the Holdridge life zone gradient and also elevational gradient. And then here is a figure that positions the data of McDonald, Cornell, and then my study all together. And the different segments you see here are the percentages of frog family found in each of our total surveys. And so as you can see, there are some pretty clear differences visually, but I found 23% more Centralidae and 11% less Cryogastridae than Cornell, but found very similar amounts to McDonald, which was interesting. And then I found about 15% less Rhonidae than both McDonald and Cornell. And then my study and Cornell's found 14% more Hylidae than McDonald. So as we can see, it all kind of differed. Similarities between some being different than mine, and then mine being more similar to others. It was just a little bit dispersed in terms of differences. And then just very quickly, I found 521 individuals, as I said, but both McDonald and Cornell found roughly 140 individuals, which was very interesting to me since I did the exact same amount of time in each site, and we all utilized Eladio's expertise. And so I was very curious why I found so many more individuals. But yeah, this was basically the distribution across the families. And then lastly, this is a very important figure because it shows how the species richness correlated with the elevational gradient. So as you can see, most of our linear regressions showed a regression as elevation went up in terms of species richness. And in terms of McDonald in my study, we both had R-squared values of around 0.5, which indicates that the points in our data felt closer to the linear regression model than that of, let's say, Cornell, who had 0.08, meaning that her data was more skewed from the average than McDonald's and mine, which essentially just means McDonald and my data found a bit more of a clear relationship between elevation and species richness. And then, just to get into why there might have been the detected differences in our projects, precipitation is a key factor in determining a neuron activity because when it rains more, there is much more clear neuron activity. And so based on the unique precipitation data of each of our study periods, that could have accounted for the different frequency of certain neuron families and also the difference in the amount of individuals. And also the trend for species richness to decline as elevation was increasing is very common in herpetofaunal studies, which basically means the elevation being correlated with the species richness is a very common finding among other neuron studies and other amphibian studies. So that was very interesting. And then overall, my data being more similar to McDonald's was very interesting because I was assuming since Cornell's was only a year prior, it would be more similar. But overall, it can kind of be a testament to the boom and bust cycles of neuron populations and the way in which they can fluctuate non-chronologically, which was contradicting to my original prediction. And then lastly, to conclude, global neuro declines have been reported since the 80s, which is not entirely understood. There are multiple factors such as changes in precipitation data, climate change, habitat destruction, and diseases such as chytridiomycosis, which is a disease that has been found to really impact amphibian populations. And so in general, expanding our understanding of these populations and their distributions can allow detection of changes in their populations, which is important as they are indicator species. So being able to detect changes in their natural distribution and richness can help indicate changes in the ecosystems they come from. And so yeah, continuing this research is very critical. And then acknowledgements. Thank you, Eladio, for all of your help. I could not have done the project without him. And then Fede, thank you for being awesome. And thank you for teaching me how to do numbers, and to everyone else who came and had fun on the frog marks. It was awesome.
We have about 3 minutes per question.
And if anyone's wondering, this is in plexus.
Oh, we know.
This is what frogs do, but they're not actively mating. He's just like really hugging her until she lays her eggs, and then he'll fertilize them. It was so cute. Question mark.
Why does a neuron
same trend. I think the higher you go in elevation, there can be for sure climates, but I don't fully understand. I would need to do a little bit more research, but the lower elevations seem to foster higher species richness, and it could be related to precipitation data and also just the characteristics of the holding life zones. But also in the future, we could use more sites in the higher elevations just to make sure the data isn't skewed that way. Thank you. Yes.
It seemed kind of like you were expecting the populations and species presence to be similar. Why would you say that we in general have that expectation when in fact maybe the normal thing is lots of fluctuation?
Yes, that's a good question. So you're asking why I was predicting it would be similar to Cornell?
Yeah.
I think I was assuming that the responses to climate changes in the ecosystems would increasingly develop as time moved forward in chronological ways. And so I wasn't even thinking about the fact that there are very pronounced boom and bust cycles among a lot of populations. And so yeah, it just didn't come to mind when I was making my initial prediction. But you're right. I think I was wondering if the boom and bust would then have a trend over time, even though it was fluctuating, but there would be on average less or more. But it was interesting that McDonald's and my study were very, very similar.
Thank you.
Antonia?
Maybe I missed this, but I think at one point you talked about Kitcha, the disease.
Yes.
Did you notice any correlations between a couple of years ago when the populations were recovering a bit more versus?
The big dip in Aneurin populations based on Cryptidiomycosis was in 1987. And so I feel like all three of our data sets were done so far away from it. It wasn't comparing from 1990, 2000, 2010. So I didn't notice anything based on that, but I bet if I did more research into surveys done right after, that would be a really, really interesting comparison.
Gracias, Elena. So, waiting for this connection of computers, remember that questions that you listen during the symposium or comment could be very useful or are going to be useful in your discussion, or please have them take advantage of these new ideas coming today. So, our next speaker is Lucas Carl Bailey from UC Santa Barbara, and he's going to speak about some night creatures that he likes to observe.
Thank you for the introduction, Fede. As said, my name is Lucas Gady. I'm a third-year undergrad student at the University of California, Santa Barbara. And today I'm going to be talking about the distribution, abundance, and habitat selection of ferruginous pygmy-owls in lowland tropical dry forest. Alright, just a little bit of an introduction and more about my study species. The scientific name is Basidium brasilienum. It is widespread in its range, known to inhabit areas from southern Arizona and Texas in the United States, all the way to northern Chile and central Argentina. And there are 15 identified subspecies across this widespread range. It is known to occupy a variety of different habitats in Costa Rica and throughout its range. And it is primarily crepuscular in its activity, meaning it is most active during dawn and dusk, but also known to be active during the day and at night. And it has a broad diet of consisting of insects, small birds, and lizards. And a little bit about why this was interesting to me and why I feel like it's relevant. We lack sufficient data on this species primarily as we head more south in latitude into neotropical areas into South America. And this lack of information may represent weakness in knowing proper conservation action and distribution, and just knowing where these owls are establishes population viability and also how strong they will be in changes or how resistant they may be to that. Here is a photo of a Frugious pygmy-owl. This was one that I found just about 1 kilometer north of Wahine Keel, and thank you to Frank for letting me use his camera to get that. And so my central question emerges as what is the distribution, abundance, and habitat selection of Frugious pygmy-owls in Wahine Keel, Costa Rica? Just to be brief with my methods, I selected 6 transects, and these were surveyed on 9 nights during mid-May, and all of these transects were sort of centralized around the town of Wahine Keel. There were geographical points every 250 meters on each transect, where I would first approach a point. I would listen for 2 minutes to see if there were any owls calling already, and then if not, proceed to play a playback or like a prior recording of a pygmy owl vocal display to elicit a response, and this is how I detected owls. I managed to see maybe 10 of them or so, but they were most auditory detections. And just a quick note, this transition from dry to wet season during mid-May coincides with the breeding season of this species in Costa Rica, which may have affected some of our results, and I will dive more into that later. Here is just a nice visual of the transects. You can see the yellow transect follows the main east to westward running road through the town of Wahine Keel, and I tried my best to sort of encapsulate a variety of different habitats through each transect survey. Alright, and for my results, I recorded pygmy owls in 71 out of 94 points along these 6 transects. This gives me a detection rate of 76%. And they were most detected in the following habitats, and these were 75% forested areas with a majority of small trees, and this was 100% detection rate, and 5 out of 5 points of this habitat type had an owl. 50% forested areas with big trees. This was also 100% detection rate, 8 out of 8 spots. 100% forested areas with small trees. This had an 89% detection rate, 8 out of 9 spots. And then near houses with big trees, this was an 82% detection rate in 18 out of 22 locations. Here's a table basically representing just that. I know it might look a little bit daunting or overwhelming. But essentially, on the left side here in the rows, I have simply whether the owls were there or not, so owl detections or no owl detections. And the columns across the top are the different habitat types. Just to quickly sort of summarize how I classified my habitats. It was essentially habitats H1 through H5, where H1 is 100% forest, H2 is your 75% forest or 25% pasture and open area, H3 is 50% forest and 50% pasture, H4 is our 25% forest, 75% pasture, and then H5 was near houses. And in order to sort of add the age, this kind of mainly considers with coverage, so I also wanted to assign a factor that could tell us how old the forest was, or at least types of trees that were there. And to do that, I added an S, which indicates more than 50% small trees, or a B, which indicates more than 50% big trees. So you can see the highlighted columns indicate the habitat types with the highest rates of detection that was in the prior slide. And now to kind of address the question of abundance, I have average owl density across each habitat type here, where on the y-axis you have average owl density, and this is owls per unit area. And unit area I defined as around 5 hectares, or the area of a circle with a radius of 125 meters around an observation point. So you can imagine that if I have 2 points and they're 250 meters apart, that with a 125 meter radius, I can end up with 2 circles that have borders that sort of brush each other, and this was kind of the most efficient way that I found to do this. And so, again, you can see on the far left, we have our 100% forested areas with small trees, with a good amount of owl density, around 1.6. And then in the middle there are 50% forested areas with big trees, and on the far right are near houses with big trees. Those are kind of the 3 distinct peaks here. And here to kind of answer the question of distribution, this is a map of the area where red dots represent an individual pygmy owl. So you can see towards the south, kind of a hot spot there with high density, and a little bit more sparse to the north and to the east, as these were habitats that were maybe less favorable. And now moving on to my discussion, where were pygmies least detected and least dense? And this was near houses with small trees. You can see I put the owl density there and unit area in parentheses. Next are 75% forested areas with small trees, and our 100% forested areas with big trees. And where were they most detected and most dense? This was 50% forested areas with big trees, with our highest 1.88 owls per unit area. Near houses with big trees, and 100% forested areas with small trees. And why might this be? Here are some of my speculations and ideas that suggest like why owls were in a given area and maybe why they weren't. The first being that they require semi-open areas to hunt, and this could be represented in the high distribution and density in the 50% forested areas with big trees. And also, moving on to the second point, they require big trees to nest. This could be why they were often present around habitats with big trees. Another idea is that the lighting from houses may attract insects, and this could be why owls are congregated in these areas of high densities to then feed from insects that gather around the lights. I didn't have a chance to display a table of this, but I basically also evaluated where the relationship between where pygmy owls were and where other non-pygmy owls were, and sort of evaluating the relationship to see if the presence of another owl type would affect the probability of a pygmy owl being there. And I concluded that pygmy owls had a pretty high tolerance of coexisting with other owl species around a given observation point. And another factor to consider that could have been attributed to my high detection rate was that the study was carried out during the breeding season, and this likely increases territorial behavior and vocal response to my playback. And as I was kind of researching this, I noticed that there were sort of lack information surrounding the differences in the subspecies, primarily physical differences in their morphology, behavioral differences, as well as differences in vocal patterns. So I kind of noted that there to just kind of as an area of future research that was evident to me as I was doing this project. I thought it was interesting. Here is a photo of a pygmy owl in a cavity nest that I again captured with Frank's camera. I talk more about this in my paper, and if you have any questions about the nesting process or some of the nesting behavior that I observed, feel free to ask me. But yeah, another great shot. And acknowledgements, thank you to Frank and Fede for all of your mentorship and teachings, and to Antonia, Vivian, and Alicia for your help in the field.
We have time for questions? No?
So what kind of like behavior did you see the owls like vocalizing with each other, like same species or different species? You notice any type of like social interaction
Between the different owl species?
Yeah, or with the same species.
I didn't, as far as between species, I just found that both were fairly vocal. There were times where the larger owls, like model owls, kind of, I think, might have negatively affected the sort of the willingness of the pygmy owls to be vocal. But I didn't actually notice any like large differences that, like, if one owl was like repetitively vocalizing, like if that really negatively impacted the pygmy owls. I wasn't sure. And then as far as between the pygmy owls, between themselves as a species, I definitely noticed some cool vocal patterns around the nest when I was watching, like contact calls between the male and the female. And then also if I was particularly close to an owl along like a transect survey, I learned later that the females give a sort of like distinct alarm call, which is different than the like repetitive hooting calls that would be typical in species. Yeah. Yes?
What does it sound like?
It's kind of not going to demonstrate, but it's basically around 15-second repetitive hooting that are kind of like conservative with, as far as avoiding counting the same individual twice. I think strategies that I took to prevent that were, I think, being with someone else on, let's see, 5 of the 9 nights I was with somebody, and having sort of a second-year person to be there and verify what I thought about whether it was a different individual or not was really helpful. Another thing is I did 7 out of the 9 nights I was either walking or biking. So this kind of allowed me to, as I transitioned between points, to like hear an owl in one direction as I'm sort of heading away. And I think that allowed me to sort of accurately judge, or at least help me identify whether the individuals were different or not.
And you said that you went back sometimes. No?
Yes, that's another important thing to know. There were several occasions where I would hear an owl at a given point, and then go to the next point, and 250 meters was like about the maximum range by which I could like barely hear the other owl calling in the distance. And I would carry out my procedure at the point, I would play the playback, I would have an owl come, and it would kind of be from a similar direction of where I heard the other owl, so there were several instances where I wasn't sure. And I would actually, 5 of the times, I biked back to the prior spot to see if the first owl was calling. And all the 5 situations it was still calling. So I think that also helps.
They're very familiar.
Strengthen, yeah.
There's another question.
Yes.
Do they have different sounds than the one that you just mentioned? Like, is there a different sound to them?
Yeah, it's generally that sound when it comes to territorial displays. I noticed that in the contact calls of the owls that I observed around the nest, those were often shorter, so 7 to 10 notes rather than 10 to 15, and they also were much lower in volume in comparison to the territorial calls.
Gracias. Do you have a brief question?
I have a brief question. So because you went out on like so many nights, what was your favorite non-creepy owl that you saw or observed or heard?
I think, that's a good question. It would have to be, thank you.
We have a liar.
It would have to be probably the spectacled owls that I heard with Frank when I did sort of by the Rio Murcielago that we all went to. That was the first time I heard those owls, so that was really cool. And the other time would probably be with you, where I thought we had the crested owl, but we weren't sure. But that was still cool. Yeah.
Bueno, thank you. I would like to welcome our next speaker. He's not bad. Elizabeth Avila from UC Santa Barbara. This is an interaction among owls. Okay. Elvin is going to mention about pumas, and if you're wondering who is around walking, observing the stars at 3 a.m., besides the mountain lion, is also Edwin.
Hello. Okay, so my presentation is about pumas in Monteverde. Where in our communities do pumas like to go and why? So let's go back and step back a little bit. My idea comes from conservation and how habitats are shrinking. And so large predators, large animals, they need large spaces. So more likely than not, when those habitats are shrinking or fragmented, they're gonna spill over and go into our areas, into our communities. So that means we need to learn how to coexist with them. We need to learn how to live with them and make sure that they don't get hurt, that we don't get hurt, and that they have their own spaces where they feel safe and where we have our own spaces where we feel safe. So it's important to know where these pumas are in relation to our communities and how and where they're spilling over into our areas. So here are the questions. How do we learn to live with them? We obviously need to know where they're coming from and where they're showing up, at what times, and what frequency. Are they showing up in our backyards or whatnot? I know, just for example, in Los Angeles, we had a very famous mountain lion called B-22 that was in Griffith Park. And he would show up in people's backyards, and he would attack people's pets and their dogs. And in fact, there was a famous story about him actually getting a koala from the LA Zoo. And the good thing is that the viewpoint of LA in general and the zoo was that it's our fault for the koala dying at the hands of the puma. It's not the puma's fault. So it's that kind of viewpoint that we need more of, so that we can maintain safety for us and them, and our animals, of course, our pets. So my way of really going around conservation is to be non-invasive. And one of the best ways to be non-invasive and study animals is camera traps. So with camera traps, you're not actually touching the animal, you're not abducting them, you're not tranquilizing them, you're not messing with them, you're just putting a camera in their environment and trying to capture where they are, where they might occur, where they might show up. And with the cameras, they record video, you can take photographs, they last for a very long time. So you have a lot of advantages with these camera traps. The camera trap that I used was the Bushnell from 2012, I believe. It used 8 batteries, a lot of batteries, and that's what the inside looks like. It uses a memory card, and that's what I use. And I set up camera traps here at the Monteverde Institute. I also set up camera traps at Rio Chante, which is like a 2-minute walk just down that way, if that. Here's a map of where the camera traps that I set up and the camera traps that other people or other organizations set up. So there was a private property that belongs to Jim Wolf, and that's where some of the camera traps were. There's Green Paradise, another private property where there were some camera traps. These were given to me by Danny from Monteverde Institute here. These are the camera traps that I set up, Monteverde, Rio Chante, FJ's house, that's Frank Joyce. He lent me his data camera trap right like a 1-minute walk away from his front door. And then BEN, Bajo del Tigre, Bosque Eterno de los Niños. There was camera traps there. I had there was 2 camera traps, one in the Jaguares Trail and the other one in the El Nino Trail, I believe, if I remember correctly. And then, of course, Naomi's family in the Gwynn Farm, she lent me her camera traps. And these are the locations. They're mostly clustered here. And then there's the one exception right out here, which is further away from Santa Elena. So I strapped the cameras to the trees. They were at eye level initially, and then I adjusted them to knee level for privacy's sake. People were complaining. So there's that. And here's an example of a camera of a puma that I spotted here. It was actually just a 2-minute walk away from here, just up the road. So they can get pretty comfortable coming through to our communities and environments. This was at 2 a.m. by the way. So no one's around that there. Here are the data collected that I was lent, not the ones that I put up. So what did I do with the information, the photographs that I got from the camera traps was I put them into the memory cards, I put them into the computer, and then I signed up for this website called Wildlife Insights. They use AI machine learning to sort through the cameras, to sort through the photos and identify what creatures, what species are there, which ones are blanks, which ones have people, which ones have traffic and vehicles. And so that's what I did. It was very useful and efficient instead of going through them one by one. And this is an example of what it looks like when it's when you're examining the photos. So it does suggest what it might be, but it asks you to identify and to confirm what the AI thinks is there. And it puts a box where it spots the animal, it tells you the class, genus, family, order, species, and it tells you the confidence level. The confidence level of what it identifies, 89% for that. And that was a jaguarundi, not a puma, but it's interesting because I didn't get many pictures of that species when they're supposed to be more comfortable around it. So interesting, but yeah, there it is. And then what I did was, because I wanted to identify the relationship between anthropological influences like buildings, light sources, and stuff like that, cars and noises, as a substitute because I couldn't find a high enough urban density map at a sufficient scale where like I can pinpoint where the camera is and it tells me, oh, this is how dense it is. I didn't have that data. That data does not exist. So what I did was instead I just mapped the nearest road from the camera location and used that as the distance between the road and the camera location as the substitute for urban density and anthropological forces. And so what I did with that data was I put it in Excel, I used some statistical analysis, R-squared, the line of best fit to see if there's a strong enough correlation between the quantity of photos of puma spotted in the camera traps and the distance away from the road. So these were the results. There were 154 photos of pumas. Now it's important to note that there's limitations with camera traps. You're not, the camera traps are not going to tell you the quantity or abundance of a certain species unless you set it up for a certain length of time and in a certain way. I didn't have that opportunity to do that, and I'm still learning. It's a complicated process that I still have to learn, but from what I was able to gather was this. I was able to see the data by year, by month, and location. And a better way to see those results and give you a bigger idea of what those results are saying is that the Bosque Eterno de los Niños in November of 2023 had the highest amount of photos of pumas. And then the rest, second, is the Gwynedd Farm. And then just the one spotted puma that I set up in my camera, which was for a total of 6 days or so. There's 3 photos of pumas, which was that one individual that happened, that one occurrence. Okay, so here is the trend line, the statistical analysis. Bottom, the further you go to left or your right, is the distance and quantity, of course, going up. So, as you can see here, the Bosque Eterno de los Niños had 73, and the distance away from the road was about 600 meters. So that's why it's up there in that corner. Now there is a data point here that's far away from everyone else, and that's the 69 of the Gwynedd Farm. So it skews the data and it skews the line of best fit, makes me have a slow statistical value far away from 1. You want, the closer it is to 1, the higher the correlation. So I got rid of that point, and now I get a higher value here, 0.8, and so now you can see the trend. And the context behind getting rid of that point was that basically that road that's next to the Gwynedd Farm, that's right next to it, there's barely any traffic that goes through there. So using that contextual knowledge, it can explain why it didn't fit the trend line. Okay, so here is the photo again of the puma that I found in here. Here is another photo from one of the private properties of puma. And the discussion basically is, what can we do to better coexist with these animals, these large predators, which can sometimes incite fear. We need more knowledge, we need more data. We need to learn how to behave with them if we confront them. But most of all, just knowing how to coexist is important, and to learn how to coexist, we need more knowledge, more data. Okay, and these are my acknowledgements. This is Alka, she's the director of the Bosque Eterno de los Niños. Maverick, who let me borrow a hard drive. Daniela from the Monterrey Institute lent me quite a bit of photos. Patrick and Mario from the Rio Chante let me set up their cameras over there. The Monteverde Institute, of course, because I set up cameras here. The residents of Monteverde, who often walk through where that puma was going, and there was a lot of traffic going through there. Naomi Solano and the Gwynedd family, Federico, Frank Joyce, Emilia, and someone I did forget was Luisa, Federico's wife, who let me a lot of data. And then we have a puma here on the tree. Here we have a video, quick little video. Puma walking through there, and then one more video. any of you else with this? And this is thanks to the conservation lead for supplying the video before from Danny. Thank you for supplying those videos, and that's the end of my presentation. Thank you.
We have a little bit less than a minute for questions.
Yes.
How many individual pumas do you think are reflected in these?
It's really hard to say because you cannot differentiate individuals. Unlike jaguars and tigers, because you can spot their spots are very unique to them. Unlike with the puma con color, which con color means same color all around, it's a very difficult. There was a study though that was actually looking at face and identifying individuals by the face. But that's a very new technique. So hard to say. I know that the population numbers aren't that high. I can't remember the exact numbers, but I don't think there's many. Yeah.
Do you have any theories about whether that such large number of photos by Naomi Stanley's farm could have had to do with even though that it was close to human populations, there's also a lot of farm animals, or is that not do pumas not tend to have as much of an interest?
Right. So there's the fact that the road wasn't very traveled. That has to do something with it. And then yeah, it's further away from people, and then it's I don't know. From what I've noticed is that the fincas here, the farms here, are very close to the forest basically, and they share a lot of land. So I think that has a lot to do with why there's so many pumas that are spotted in the farm, because they can escape really easily. Yeah.
But you don't do you think there's any them choosing to be near the farm too? So those animals is that a problem?
So one thing I don't want to say that it causes them to go is that because there's a lot of farmers who have dead animals, pet owners who have pets going missing, and they're like, oh, it's a puma. And there's a lot of studies out there that say it's really hard to judge whether it was a puma or not. And from the studies I've seen, these pumas don't actually like to attack cattle unless they're very desperate and injured, because then they're easy prey. But aside from that, they don't want to get involved with humans, and they know that there's people there, so they're going to avoid those places.
Thank you.
Yeah. Thank you.
Some ideas might come to your mind straight away, like if you were to see someone crying, you might think that they're sad. Or if you saw someone smiling, you might think that they're happy. But what would you say if I was to ask you to describe the behavior of this guy? Or her? Again, you might have some ideas just looking at the body language of these two individuals, but generally it is much harder to understand the behavior of other animals that aren't human. This exact uncertainty is what inspired me to design a project that would allow me to better understand the behavior of different animal species. Specifically, I was interested in the behavior of bats. So I have three main questions that guided me through this project that I was interested in answering. The first being, how does the behavior of bats vary within and across species? Is there any differences in the behavior of bats versus male and female? And is there a phylogenetic trend in any of the behaviors that I was noticing? So, my methods for this project, to capture the bats that I collected data from, I set up a few mist nets every night that I collected data. They were open for roughly 4 hours, and every 15 minutes I would check on them and see if a bat was in the net. If a bat was in the net, I would then very gently and politely remove it from the net and place them individually into a cloth bag
checking all the nets, come back to a little central processing station set up at each study site, and go through some standard data collection from each individual bat. So I measured the weight, I also measured the forearm and tibia length of each individual bat, and then identified factors such as their biological sex, what species they were, and also their age. And it's not on here, but the bats were also marked, so to prevent recapture and collecting data on the same individuals. So to quantify the behavior that I was noticing in the bats, I implemented a 5-level scoring system that took into account different physical displays of aggression. So essentially, each increasing level from 1 to 5 signifies a higher level of physical aggression from the bat. So level 1 is a score I would give to a bat that was super calm, not moving. 5 is a score I would give to a bat that was just going crazy, biting the handler or moving their wings a lot. So each bat ended up receiving 3 scores. The first score was given just off of initial observation. So the bat was in the mist net, no one touched it, and I gave a score based on how it was acting. The second score was an average of 4 scores throughout each stage of handling. So, like I mentioned, weighing it, measuring it, identifying its species, and all that jazz. And then score 3 is essentially just an average of score 1 and score 2. So it takes into account initial observations as well as observations of behavior while handling the bat. And score 3 is what I used in my statistical analysis. So, after collecting all of my data, I was able to run some statistical tests that allowed me to compare behavior score 3s across like a bunch of different comparisons. So the first comparison that I made was between 2 species, Curlia sowelli, and the other one was Dermanero toltecus. So in this figure, the x-axis has the species names, and the y-axis is the average behavior 3 score of individuals. So as you can see on the left, the Curlia sowelli had a higher behavior 3 score, signifying a higher level of aggression than Dermanero toltecus. And upon statistical analysis, it was actually found that this is a statistically significant difference in behavior scores between these 2 species. The next comparison I made was between male and female bats. So this is a pretty similar graph to the one before, except on the x-axis now it's male and female being compared instead of 2 different species. And on the left we have the female distribution of behavior scores, and on the right we have male. There is one outlier in the male distribution score. However, the female behavior score was lower on average than the male behavior score. And when I ran some statistical tests, this was in fact found to be statistically significant in terms of difference in score. And the next comparison that I made was between different subfamilies of bats within the broader family Phyllostomidae, which if you don't know is the group of beak-nosed bats. So I, in total, did analysis on 3 subfamilies. 2 of those subfamilies didn't have any significant difference in their behavior scores of the species I was comparing. But this family subfamily in particular, Stenodermatinae, had some interesting distribution across the species that I was considering their average behavior scores. So on the x-axis, again, we have the species names. The y-axis is the average score 3. And there are a couple outliers to point out. There's one in the species Dermanero toltecus right here, another in the species Patyrhinus vittatus, and another outlier in the species Sterina hondurensis. So, upon statistical analysis, I compared these behavior scores all to each other and found that there was statistically significant differences in some of the behaviors of the species. Particularly, one species in common was behaved differently than 2 other species, which was Patyrhinus vittatus. This species here behaved statistically different than Dermanero toltecus, this species here, and Centuria sinex, this species over here. So, using all of these results, I wanted to spend some time talking about what they mean and some possible explanations for the differences in behavior I noticed. So, to the first point, Curlia sowelli were found to act statistically more aggressive than Dermanero toltecus. And I attributed this to 2 main possible explanations, which is diet and lifestyle. So as mammals, a lot of our energy is influenced by our diet and the food neotropical bats have found that these two species in particular, Curleus sowelli and Dermaneratoltechus, do have different diets from one another. So they're both fruit-eating bats. However, they eat different kinds of fruit and they eat different families of fruit. So in that respect, I thought it could be an interesting analysis to think that perhaps the diet is influencing the behavior, and the food that Curleus sowelli is eating is giving it higher energy and making it act a little bit more aggressive than Dermaneratoltechus. And also the second point is just lifestyle, because these are two different species. So there's factors within bat colonies that could be influencing their general behavior, such as just social interactions between one another. The next comparison I want to talk about is the fact that males are found to be more aggressive than females. Again, there's a bunch of different factors to consider when thinking about why this behavioral difference was noted. The main ones that I focused on in my analysis were differences in reproductive rules, so there could be influences of females giving birth when you were young that can make them behave different than males. Social interactions in general between males and females within colonies can be different. But most notably, I think that there might be more to know about. There might be more influence for hormones affecting their behavior, because across mammals, males typically have more testosterone, which leads to more aggressive behaviors, so it can explain this trend. And the next comparison is the subfamily comparison. The fact that consistently Platyrhynus vittatus behave differently than two other species in the subfamily just interests me. So I decided to kind of take a more evolutionary perspective look on why this would possibly be happening. This is a subfamily tree of the subfamily Sturmatinae, and this point right here represents the common ancestor of the subfamily, and down here is where Platyrhynus vittatus is. So the Platyrhynus genus in general is actually the most distantly related genus of this subfamily, whereas the two other species that were found to have different behavior than Platyrhynus vittatus, they're much more closely related to the common ancestor and each other, and in general the other species within the comparison were found to be more closely related to each other. So intuitively, like the other two subfamilies that didn't have behavioral differences with each other, you might think that subfamilies would behave similarly to each other, or species within subfamilies would, but this evidence shows that there is still variability in behavior within subfamilies that could be attributed to evolutionary factors like shared evolutionary events, genetics, or how distantly related they are to one another. So in conclusion, my project showed me that there is just a lot of variability in bat behavior and personality. There were a lot of outliers in some of the comparisons I made, which I thought were super interesting and just showed that not all individuals in the species will necessarily act the same as each other, but I was still able to draw some interesting conclusions about general behavioral patterns. The cool thing about my study, I think, is that my information and results are quite unique. In fact, I couldn't find any other scientific literature that did the same methodology or found the same results, which is cool, but also makes it hard to make definitive claims about the results that I found. I think some future research that could be really beneficial is doing a study similar to this again, possibly catching more individuals to add to comparisons that I made, and see if there's any differences that happen when more individuals are included in the study. Or another thing I think would be really cool is if another character trait was used or was quantified in the study. So I used aggression in my study to quantify my analysis of behavior, but maybe some scientists in the future could find another character trait to quantify and look into, see how that affects the behavior of other animals. So really quickly, I want to thank my primary advisor, Federico, and my partner in crime, Anna. I could not have done this study without you guys. Thank you for bearing with me every night. My secondary advisor, Naomi, everyone who allowed me to collect data on your properties, thank you. Couldn't find bats without that. The Monteverdi Institute, thank you for providing vehicles to get to our study sites, and Adriana Arias for providing just unwavering support and guidance.
We have a little bit less than a minute for questions.
I remember when I would go back with you, we were looking at the, if it's cartilage or bone in the wing to determine age.
Yeah.
Did you ever look at age as an impact?
I wanted to really look at age, much more similar to like the adults of Curleus sowelli than the adults of Dermaeroteltechus, which I thought was super interesting, and I would have loved to have done more comparison and analysis of that, but unfortunately I did not catch enough juveniles in total to make like any kind of significant analysis or comparison. I think like in total we caught like 7 juveniles out of like the 74 bats that I ended up catching, so it would have just been kind of skewed analysis, but that was something I was interested in. Yeah.
Gracias, Emma. So a couple things to say. There are chairs here in front. Please come in. Welcome. Thank you for coming. And the other thing to say is Emma, the speaker, is going to stay around for a couple hours. If you have more questions, you can meet her. As you can see, more people is coming to the auditorium because we are close to the most interesting presentation of the Thank you for coming. So this is going to be the last presentation of this second section. After this, we can lower the score of aggressiveness in personality. We will have more coffee, sugar, tea, sugar, candies, and things. You know? What?
No.
From UC Santa Cruz, they will speak about bat species diversity and compositions in the region of Monterrey. Thank you all.
Okay, so as Federico mentioned, I did my study on bat species diversity and composition along an elevational gradient in the Monte Verde region in Costa Rica. So there are 112 bat species in Costa Rica, and they play important roles in forest regeneration by pollinating and dispersing seeds. Bat reproductive rates are pretty low, and so if their distributions get too low, it's very hard for these species to come back. And they are potential excellent indicators of climate change because they're very sensitive to environmental cues and factors. Climate change is affecting bats as well as their distributions, and bat species have been documented at higher than normal elevations. This suggests that they are adjusting to warmer temps, and this brought me to my central question, which are bat species changing their distributions and compositions due to climate change? Okay, so I looked at 3 different elevational sites: San Gerardo at 1,200 meters, Bajo del Tigre at 1,350 meters, de Estación Biológica at 1,550 meters. I misnetted for 4 nights in each location, and I set 3 misnets at each location. So here's a little example of a misnet. You can see it has these different things that go across, and then the bats will get caught and they fall into little pockets, and this is actually where they get tangled. I would check every 15 to 20 minutes, depending on how slow the night was, and then I would carefully untangle the bats from the nets, and then I identified the bats using the key for bats in Costa Rica. And I was very closely following the methods done back in 2003 by another student named Amanda App. So here's a video of us untangling, well, Emma and Federico untangling a bat from the net. It was chewing on it, and so it was a lot harder to get it untangled, but you can kind of see how they get caught in the net, and then a lot of work is done to get them out. Federico doesn't eat gross. Sorry, Federico, I'm calling you out here. Here is a video of measuring the forearm, which Emma mentioned in her presentation, but you can see the It's just like a standard ruler. We just measured it in millimeters. And this helps us identify most of the species that we use. Look at his little tongue.
It's so cute.
And then lastly, here's a video. This is Centurio senex, the hairless-faced bat. And you can see this one here, and this one down here. And this is after we released them. We set them on Frank's things outside his windows. Waited until they flew away. Okay, so this is just my number of individuals I caught. And I caught 92 individuals in 18 species. And I caught the most here in Bajo del Tigre at 49 individuals, and the least at the Asociación at only 17. So this is looking at Shannon diversity, which we all learned about. And if you guys remember, H prime is the number of species and the number of individuals of each species we caught. And then H prime max is if we caught the same amount of each individual of each species, that's the max diversity that we get. Another very important thing for my study is habitat zones. So habitat zones in Monteverde correspond to changes in fauna, which are animals, and flora, which are plant compositions. And I looked at zone 2, zone 4, and zone 5. So zone 2 is Bajo del Tigre, zone 4 is Asociación, and zone 5 is Santa. So a couple of definitions before we move along. Abundance is the total number of individuals of a species in a given area, and the distribution is a range of the species. So this is kind of how I broke up my data. Abundances are defined by uncommon, common, abundant, rare. And so if abundances were going down, it was going from like abundant to common, common to uncommon, or uncommon to rare, and then vice versa for if abundances were going up. As I mentioned, 2, 4, and 5 were my distributions. I compared abundances and distributions from 2000 to 2024. The study done in 2000 was done by Tim and Laval. In 2024, obviously, was done by me. Okay, so San Bernardo, Corolla, Crispicillata, and Glosophaga commercialisi were not previously found in this location. Anura geoffrey, Germinura totekis, Pelurrinus vittatus were previously found and now were not found in San Bernardo. Corolla sewelli, Lontrina aurita abundances went up. And then Myotis pilositibialis, Sternera hondurensis, and Sternera mordax abundances all decreased in San Bernardo. And this is a picture from Fede back in 2015. And I was very happy to find that on Twitter. Bajo del Tigre, Pelurrinus, Hillary, and Germinura watsoni were not previously found in this location, now they were. And then Artabaeus homaicensis, Hylonectarus underwoodi previously found, and now they were not found in this location. These guys, the Myotis riparius and Corolla persiflata abundances increase in Bajo del Tigre, and then Anura geoffrey, Myotis pilositibialis, Pelurrinus vittatus, Sternera hondurensis abundances both all decrease. And then lastly, for the Asociación, Corolla castanea and Pelurrinus helveri were not previously found here. Anura geoffrey, Glosophaga commercialisi, and Sternera mordax previously found. Now, when I did it, was not found. They were not found here. And then, interestingly, there were no species with a higher abundance in the Asociación, which is where I thought I was going to find. Instead, Artabaeus homaicensis, Corolla sewelli, Germinura totekis, Hylonectarus underwoodi, Myotis pilositibialis, Pelurrinus vittatus, Sternera hondurensis abundances all decreased. I know, I practice these a lot. So, decreases in abundance, it means that the bat species are moving away from these elevations, while increases in abundance
abundance means they're related to it. Interestingly, my Shannon diversity was very similar between all three of the sites, but it has increased significantly over the past 21 years of the Symposium, meaning there are more species moving up, even if their abundances were lower, there's still overall more species found here. There are a number of other factors that are possible contributing to these results. One major one is a lot of reforestation has been happening in the Monteverde region. Bats live in certain types of forests, and so as reforestation is happening, bat species might temporarily move to a certain forest type and then move out of it, and so maybe we were finding some of that. Another big thing is urbanization affects bad biases and distributions since they don't like a lot of light, and so they move away from regions of urbanization, and Monteverde, once again, in the past 24 years has a lot of urbanization has happened here. So it's very important to track the changes in bat distributions and abundances, especially as climate change is affecting a lot of things. And continuing studies like this in the future will ensure the conservation of these animals. I'd like to acknowledge Federico, my Batman, Emma, my Robin in crime. I don't know what I am, but that's okay. Frank, for letting us use this yard. Naomi, the Sacramento staff and associate staff.
Thank you, Anna. I am here. But now we have time for questions.
I don't know if you did a ton of research into this, but I'm curious, what makes bats sensitive to their environments, like making them indicator species?
I read a paper that was talking about a lot of different factors that they could be sensitive to different things. And I can't remember the exact results, but they were finding that bats respond to different stimulus a lot faster maybe than other animals do, and so they're potential good bioindicators, but it's not like 100% different.
Yes?
If you would do the study, then what would you do?
More locations. If I was only able to look at 3 zones, I would love to have looked at all 6, which obviously is not possible in 3 weeks, but looking at all the different elevation, like all the zones, just to see the overall. Because the Tim and the Wall one, they looked at all 6 zones, and it was a very complete study. So I would like to look at all 6 zones. If I had more time.
What was your favorite bat?
The nectar feeding bats, like the Glossophaga one with the long noses. They're very cute.
You might have said this, I might have missed it. How many changes did you detect in the sense of a new species being a new species in a new area versus one not occurring where it had previously occurred? I mean, just más o menos, like, you know.
I say about 3 per. So like, on average, I say I found 3 new species and then also 3 that are not in there yet.
For each site?
Yeah.
Wow, okay.
Obviously something more, but I think that.
Cool, okay.
One bat that they call the wrinkle face bat, usually you see 1 or 2 per year, but in France we call like 7. So there is like a nice colony of wrinkle faces.
I haven't. Somewhere in here. Here. Okay. Since you're descendants.
Usually it's rare and it's still being rare, but in your house it's common.
You have one? Yeah.
But
Like me.
thank you for
What?
how much time?
I think we should go to 1 o'clock. But very important, for people in the afternoon sessions, please come up and make sure that your presentations are either on your computer or on my computer, okay? So don't wait until like 3 minutes before you're supposed to talk to put it on, okay? There'll be a little bit of a line for lunch, and of course, todos los invitados están invitados para almorzar.
Right now?
Yes, right now.
Cheers.
Let's go.
It's so hard. You're not
Okay, no, yo entendí que la organización es como
This creates an ion concentration gradient within the pulvinus, and thus an osmotic potential. So water flows from the lower part to the upper part of the pulvinus. And the lower part shrinks, and the upper part swells with water, and this swelling is what causes the leaf to drop. Okay? And it drops in a matter of seconds. So this behavior is energetically costly to the plant because of this active ion transport. As the ions return to an equilibrium state, so does the water, and thus so does the position of the leaf, slowly in a matter of minutes, return to its original position. So the question is, or a question is, why would a plant perform this energetically costly behavior? The adaptive significance is theorized to prevent herbivory by scaring away herbivores and appearing wilted and less appealing or appetizing. Okay, so the primary pulvinus and the tertiary pulvinus can activate independently. So here depicts a leaf before and after tertiary pulvinus activation. So note the leaflet's folded, but the leaf did not drop. And then here is a leaf before and after a primary pulvinus activation. So the leaf has dropped, but the leaflets are still open. Okay, so far we've learned that the primary pulvinus activation is energetically costly due to that active ion transport. And we've seen what happens to the first activation, but what's the behavior after that? So after some time has passed, if I apply another touch stimulus, will the primary pulvinus activate again? Will it not? If it does, will the angle drop be more or less or the same? Which motivates my central questions. So there's 3 central questions to my study. The first is, how will the leaf's angle drop change with repeated primary pulvinus activated stimuli? What could explain this behavior? And how will concurrent tertiary pulvinus activation affect these findings? Okay, so to answer these questions, I followed this experimental procedure. I propped up the stems with rocks and sticks, and then I applied a touch stimulus. And the touch stimulus, if my arm is the leaf again, is a light touch on the leaf, trying not to activate the tertiary pulvinus. If the primary pulvinus activated, I recorded the absolute leaf angle drop. So pay attention for this part because it's important for the rest of the presentation. When I say leaf angle drop, I mean the angle traveled by the leaf immediately after the primary pulvinus activates from the start to the end of its movement, so that angle. So an example would be like, this would be a 45-degree angle drop, and this is also a 45-degree angle drop. So it's from the beginning to the end, okay? Great, so I did this, I applied the stimulus, then I waited 5 minutes, and then I repeated the same touch stimulus, and I repeated this for 18 total stimuli, for 60 leaves total. And for each leaf, I performed 2 experimental conditions. The first is the 18 stimuli I just described with the no tertiary pulvinus activation. And then for the second condition, I performed the same 18 stimulus experiment, but with the addition that every time the primary pulvinus activated, I also activated the tertiary pulvini with a light pinch. And so each leaf underwent both conditions on separate days. And I'll now start to discuss my results. So this plot on the x-axis is the 18 touch stimuli that I described before, 5 minutes apart. And on the y-axis is the absolute leaf angle drop in degrees. And each dot represents that leaf experienced that angle drop at the corresponding stimuli, okay? And the dark black dots correspond to the mean for that stimuli, and the error bars the bars are standard deviation. And the left plot here is for the no tertiary pulvini condition, and the right is for the tertiary pulvini condition. Okay? Okay. So, this is the takeaway. The highest mean leaf angle drop occurred at the first stimulus, see, and followed by a lower leaf angle drops for subsequent stimuli in both experimental groups. Okay? Next result is in this plot, the x-axis is the same as the 18 stimuli before, but now the y-axis is the number of primary pulvinus activations for that stimulus. So again, the left and the right correspond to the two conditions. And to help orient us here, at stimulus 1, all 60 leaves fired had primary pulvinus activation, and none at stimulus 2. And the takeaway here is that the highest number of primary pulvinus activations occurred at the first stimulus, followed by, again, lower number of activations for subsequent stimuli. So let's discuss this a little bit further. Remember, my first question was, how will the least angle drop change with repeated primary pulvinus activating stimuli? The answer is the mean leaf angle drop and the number of primary pulvinus activations are highest at the first stimulus, and then for both experimental conditions, and then both of those were lower for the remaining stimuli for both experimental conditions. There's also evidence of a refractory period, so if a pulvinus is in its refractory period, it will not activate again, even if the same stimulus is applied that activated it prior. This refractory period is evidenced by the no primary pulvinus activation at the second stimulus, followed by a slow, gradual increase in the number of pulvini activated thereafter. And the second question was, what could explain this behavior? So there's two main mechanisms for explaining changes in plant response with repeated non-damaging stimuli. One is habituation, which is a gradual response to stimuli, to repeated stimuli, and another is mechanical exhaustion, which is a significant and sustained response after the initial stimulus. I hope that everyone can see that this fits the mechanical exhaustion explanation. And so, takeaway from this is my results indicate a mechanical exhaustion mechanism for repeated primary pulvinus activations, potentially due to that resetting of where the ions are in the pulvinus. And then finally, how will concurrent tertiary pulvinus activation affect these findings? Overall, both condition groups exhibited the same overall behavior. Okay, lots of people and places contributed to this project, but here are a few. Very grateful. Thank you for listening.
It's time for a couple quick questions.
Can you put up a graph where you show if mechanical exhaustion was occurring, why wouldn't the angle continue to decrease? Because it goes up and down, but it doesn't stop responding. And another question I have is, I think actually now I think in the same project, so if the minus is up here, is it actually an active lowering, or is it gravitation, like gravity pulling the leaf down?
So those are two questions. It isn't active, so I explained that there's a flux of ions and there's a flux of water. And so if you have the lower part of the pulvine shrinking and the top expanding, that directly causes that drooping. So it's not that the water flushes out in its gravity. There is an active osmotic process going on. And then the second question was about, like, why doesn't it just go down to this and then stay where it goes? So there's a lot of things that I didn't talk about in my study. This wave-like behavior is statistically significant with 2 peaks and 2 troughs. I am doing this as sort of the refractory period that I talked about before, where after the initial stimulus, there's a large flux of ions and a large flux of water, and that causes a large response. And then after that, between stimulus, it may be activating, but there's not enough time for it to return to the initial equilibrium state. But it is, to abuse this word here, it's trying, right? It's trying, but it's receiving more and more activations. And so that's what's causing, it's trying, but it is being stimulated more, and so it will use water. That's what's causing that wave-like behavior.
eso es algo muy sencillo. Solo tiene que tocar esto.
No, es que se supone que se conecta y que está volumen. Sí, para eso se lo está registrando, pero debería sonar aquí porque son los ramos.
¿Y esto?
Ya, ahora sí. Perdón.
Hello, hello. Oh, this is exciting using the mic. So, hello everyone. My name is Ileana Romero, and I go to UC Berkeley studying plants. Can't you tell? I love plants. The biochemistry of plants, as you will soon see. So my project is on photosynthetic rates and pigments of setiophytes on an elevational gradient in Monteverde, Costa Rica. So a little introduction to what exactly are setiophytes. They're actually a new morphological group within biophytes. This is new as of 2018, actually. They realized that a lot of their morphology, especially on how they reproduce and their chloroplasts, are very similar. Very different to hornworts, which are the third group in bryophytes. As most of you, well, some of you probably know, that bryophytes are mosses, liverworts, and hornworts. And trichophia are like anything with seeds, including flowering plants, but also they include sporiophytes for ferns. So bryophytes are pretty much non-vascular plants. These plants do not have a very strong cell wall, so they do not grow very tall, but they are very widespread, and they are a keystone species in many ecosystems. This is primarily because of how sensitive they are to temperature change, humidity change, and even just air pollution in general, since this can pretty much stimulate how they do photosynthesis. And this is the primary reason of why I would like to study setiophytes, mostly because of how common they are and easy to find in any ecosystem. So a little bit more background on plant pigments is that there are two primary pigments. There's photosynthetic pigments and protective pigments. The protective pigments are a little bit more harder to do, so I decided to do photosynthetic. And there are two types, as previously mentioned. There are chlorophylls and karyotypes. There is chlorophyll A and chlorophyll B, and both of those absorb different light waves. So as you can see, there is the visible light spectrum, which is primarily white light. And so you have chlorophyll A, which is primarily absorbing blue-violet. You have chlorophyll B, more on like the blue-green, and then keratins go from blue to yellow, and then they also absorb again in the more red area. And because of this, when you look at a plant and you notice that their vibrancy on how green it is depends on how much pigment they have. So if they are absorbing any other color, it could be due to oil pigments or due to the reflecting of light. So this is the primary reason why we see green plants is how they absorb light. So my central question is, how does elevation affect the rate of photosynthesis and photosynthetic pigments of setiophytes in a tropical cloud forest? So that's a picture of me walking up the mountain. And funny enough, this is actually one of the setiophytes that I found. It is part of the Jungermannulides class. This class of setiophytes, specifically liverworts, are the leafy liverworts. There are three major liverwort groups, which is simple liverworts, complex liverworts, and leafy liverworts. Specifically in the thallus is how you could classify whether they're monsters or liverworts. You'll see that when I show my methods. Alrighty, so my methods. So first one was field collection. And then next I did a photo experiment. And then I did a paper chromatography. I will go more in depth on what each of the three methods do. So in my field collection, I went to the estación, and the estación is at 1,550 meters, and then I went all the way to the top of the principal trail, which was 1,780 meters, but it can go up to 1,790 meters. However, as you can see in this next photo, there was a tree that blocked, so I could no longer reach all the way up. So from starting point to end point was 1 kilometer. And so all the way at the top, so from here, which is right there, every 60 meters, I tagged a tree all the way to the bottom, and that was my study sites. There were 19 different study sites, and at each study site, I collected 3 samples. So in this photo, you can see there is a lot of greenery. This is where liverworts were. On this tree were actually mosses, and on the ground were other liverworts. And so I pretty much only gathered relatively 3 meters from where I marked the tree. And then in this site, it is number 12. And as you can see, you can find them more on the tree, on the little log, and on the grass. So I did include the different substrates in my data. However, it got very complicated in the graphs, so I'll briefly mention them in my results. So in my float experiment, this is how I was able to test photosynthetic rates. So after collection, I waited 24 hours to do this experiment, and I used sodium bicarbonate, which is baking soda, and 100 milliliters of tap water for each sample. I just made this really big batch using these ratios to simplify instead of making this over and over and over. I probably made like a gallon of this. And then I got all 3 pieces from the sample site, and then I added 30 milliliters of the sodium bicarbonate solution into a syringe, and then from there I created a vacuum by holding my thumb and shaking it. And then from there I added it into my beakers. And because of this, I was able to stimulate the plants, and they were pretty much like kind of at the bottom with the light turned off. And then once I turned on the light, that's when they started to produce oxygen and started to float. And so from there, I pretty much measured this in seconds. So for paper chromatography, for those of you who don't know, is a chemical analysis of where you add a solvent at the bottom, And then you add your paper and a pigment, and then it moves up based on polarity of the pigments. So I added all of my pieces from my experiment in float 1 per site. So this data point has 19 for each site, while the float experiment had 57 data points. I added 1 milliliter of pure acetone to make my mixture, added it 3 to 4 drops, then the alcohol will rise up the paper, and that was my end result. So in order to calculate this, I use RF values, which is where you get the line of origin and the solvent front. Most of the time it was 9 centimeters, and then from line of origin to like chloroplast B would be 3.5, and then chlorophyll A would be like 4.5, and then keratins would be like 7. And then you divide those numbers up, and you'll see the graph soon. And so I ended up collecting 18 different liverworts and 34 different mosses. And so here is the float time by seconds, and then the elevation. And then you probably notice that there's 2 distinct different groups, one at lower elevation, one at higher elevation. And one interesting thing was in the middle is where it got really curvy and really steep. So it was a like really distinct decline in elevation. There's pretty much no relationship in float time or elevation, nor in setiaphyte or elevation or float time and setiaphyte, unfortunately. But it was really interesting to learn this. Another really interesting thing is on pigments and elevation. There was also no relationship in either of them. And then the next thing is pigments and average float time, and these did have a really big relationship between them. And this is primarily because the vibrancy of the pigments and how long they were able to produce pretty much created more active chlorophyll and increase in pigments. So when I did the paper chromatography, there was a very distinct relationship on how prominent each of the pigments were. And also I'd like to thank Naomi and Federico, especially Marvin and Carmen, for using the principal trail. And shout out to my squad, especially after I got insane from all the fumes.
It's time for a couple quick questions. What made you have the idea?
A project like this? I think it was when we did the orientation trails when we first got to the station. I was with Federico, and then I noticed that there's a bunch of grains on these rocks. And I was like, what is this, algae? And then I took a picture of it, looked it up, and turned out it was liverworts. The RF value is the distance between line of origin solvent as your denominator, and then where the line of origin to pigment as the numerator. So it's a division between the different pigments. That's currently unknown. There's currently a lot of research into that as far as I know, because it's the way plants kind of use the different pigments. So there's a lot of ways plant biologists are trying to find this out by testing different light to see if they'll grow better or if they'll produce something different, or if that'll like have secondary like traits or mechanisms change.
Thank you again. of mating pairs in Argia damselflies. So to start, this is two individuals in the Argia genus. These are Argia chelata, and this genus is very, very common around Ponte Verde. You can find them in the ponds, streams, river, and a lot of them are characterized by this very blue pigment dispersed throughout their body. Now, like all odonata, or dragonflies and damselflies, these two individuals are exhibiting a behavior known as tandem. This behavior occurs before, during, and after copulation in all of the Argia genus damselflies. And basically what it does is ensures that the eggs that are laid by this specific female are the ones that are fertilized by this male. So they hold on throughout the copulation process and while she lays her eggs. Now the way this works is the male has, in all Zygoptera, or damselflies, the males have two sets of terminal appendages known as cerci and paraprox. And they use these little claspers, essentially, to grab onto the thorax or neck of the female here. All right. So, in a lot of different in a lot of different damselfly species, there's been evidence that larger individuals have more fitness. There's a few different reasonings for this. One of the main ones is that the larger the individual, the greater the fat reserves and the muscle mass that they'll have. And these reserves are going to be used during behaviors like flight, which are part of mating and hunting and male-male competition. So by having these larger reserves, it can allow them to be better flyers in general. Additionally, in many different insects, as well as a few species of Zygoptera, it's been shown that the larger the female, the more fecund they might be. This is because they'll either produce more eggs or live a longer lifespan, but they'll be able to produce more offspring throughout the course of their lives. And then finally, during this tandem behavior that we were just looking at, sometimes other males will try to displace the current male attached, and there's been evidence in certain Zygoptera species that the larger males are better at withstanding these displacement attacks. So, with all of this evidence for greater fitness in larger individuals, I was wondering if within Argia there would be assortative mating. Now, positive assortative mating is essentially when individuals look for mates that have similar characteristics to them. So in this case, it would be larger individuals, larger males mating with larger females, and smaller males mating with smaller females. And additionally, with this information about the displacement attempts, I wondered if that could be correlated to the size of the terminal appendages, and whether the larger individuals tend to have larger terminal appendages, which might help them stay better attached to the females. So, these led to two of my research questions. Does body length determine terminal appendage length in Argia species? And does assortative mating dependent on body length occur in Argia species? So for my methods, I spent 5 days circling around the Hotel Del Mar pond between 10 a.m. and 1 p.m. Damselflies and most Zygoptera and Odonata really need the sunlight to survive, so they spend most of their time out in the sun. So I was going during the peak sunlight hours of the day. And I was trying to catch as many tandem pairs and individuals as possible. Now each time I caught them, I would take a photo next to a ruler and then mark them with a paint pen so I wouldn't measure them again. And then later I used ImageJ to get direct and specific measurements of the body length in both the females and the males, as well as the length of the terminal appendages in the males. So, within these 5 days, I ended up catching 53 individuals between 4 different species. Now, when I looked a little bit closer and did some identification, I realized that over 90% of these individuals were between 2 species, Argia elongata and Argia anceps. So due to sample sizes, these were the only 2 that I ended up analyzing in my project. I also ended up catching 7 tandem pairs, so both the male and female at the same time. And all of these tandem pairs happened to be Argia anceps, which was very nice for my analysis because I had a little bit more data for a specific species. Okay, so to start, these 2 graphs, there's a lot of words on here, but basically what they're showing is the relationship between the terminal appendage length and the total length of the body. Now, when you're comparing the size of a specific body part in comparison to the entire body size, this tends to be an exponential relationship. So the natural log of both of the measurements were taken to linearize this relationship. So along this x-axis here, we have the natural log of the total male length versus on the y-axis, the natural log of the male terminal appendage length. Now, in both of these, we see that the variation of the points to the line is all over the place, and they're not really following a specific pattern. The R-squared values are very low, and they're not demonstrating much correlation, showing that in these two species, body length is not determining terminal appendage length. So this graph here shows length-based assortative, or looked for length-based assortative mating in Argia anceps. And along the x-axis, we have the total male length in millimeters versus the total female length in millimeters. And each one of these 7 points represents both the male and the female of the tandem pairs that were caught. Now again, we don't see much of a strong relationship between here. The R-squared value is very low, only 0.013. So there's not any main, it's not showing any major linear correlations here. So we can see that mate choice is not going to be influenced by body choice in these species. Sorry, it started.
Okay, sorry. So when I was looking at this, and after seeing that there really wasn't any relationship between the length of the terminal appendages and the length of the body, I started wondering if maybe the terminal appendages length would be directly correlated with the female length, and that only males with larger terminal appendages would have the ability or be more likely to be mated with these larger females. So I tested this, and there was still no linear correlation. What? Oh no, sorry. Okay. But yeah, so here it's the male terminal appendage length along the bottom, and then the total female length along the y-axis. But again, a very low R-squared value and not a strong correlation here. So again, we can see that in Argia anceps, mate choice is not influenced by the terminal appendage length. Okay, so these are honestly unexpected results to me based off of so many different explanations for why larger males and females could have higher fitness, but this also implies that there might be other factors influencing the mate choice. For example, in some Argia species, or first of all, damselflies and dragonflies in general have really good, really strong vision. It's some of the best within insects, and in other Argia species, there's been evidence that females will tend to prefer some of the duller colored males. And this is honestly, even in that research, wasn't that expected, but what they noticed was that the brighter colored males, the ones that were more blue, were getting more attention from avian predators. So they theorized that it's specifically in that species, the females were avoiding those more brightly colored males because it would expose them more to predators. Additionally, within different species here, there tends to be not a lot of variation within the clasper shape, even though there tends to be variation within the length of it, like what I saw. But there's a lot of theories that say that the shapes specifically for each species are used for species recognition when those are in tandem, and that they, as they're getting into tandem, they can, the feeling between the terminal appendages of the male and the plates on the female help them identify one another. So if there tends to be any variation within species, this might also have a role in mate choice, because the females and males would probably be more likely to choose a mate that they are expecting and that they are used to that feeling. And this is the same idea for the strength of the claspers on the males. And then finally, the location and timing can play a role in mate choice. There tends to be a lot fewer females than males on any given day. And so the males were spread out all around the pond, but females, there were just a lot fewer of them. So the choice of where the female goes is going to influence who she ends up mating with, and also the time of day that she is out and about. So, in conclusion, in Argia elongata and Argia anceps, we saw that male size does not determine the length of the male claspers. And in Argia anceps pairs, I determined that mate choices are not determined by either body size or clasper size. So this is implying that other factors may influence mate choice. And there's honestly not that much research on this genus in general, and especially, which is really interesting because not only are they a very bright colored species, they're also sexually dimorphic, and there's a lot of different reasons why it seems like they would draw in biologists' attention, but there's a lot more research to be done about these mate choices in the future. And I'd like to thank a lot Emmy Triana for all of her help as my primary advisor, as well as Noam Solano, as well as my wonderful homestay family, the Cruzabando family, and the Hotel Del Mar for letting me use their pond. Thank you!
There's time for a couple questions.
Okay. When you see them in tandem, does that imply that there is a delay or no?
Yeah, so actually I found a study, that one of the very few studies about this specific species, Argent ansets, showed that 95% of successful populations led to, 95% of successful tandems led to copulation, which was very interesting. And so it also implies that a lot of the mate choice occurring is happening before this tandem and as they're getting into tandem. Like the attempts there. And so that was really interesting. At first I was looking and trying to differentiate between the pairs that I just saw in tandem versus the pairs that I saw where the female was ovipositing. But yeah, then I actually, after the study, I discovered that like almost all tandem pairs lead to copulation.
One more question?
Does the male ever try to apprehend the female if she's moving, or do they do it like, does he apprehend her neck when they're actually alighted somewhere, or is it an in-flight thing? The reason I ask is because there may be a lot of checking things out, and maybe the female is choosing a male based on his persistence. Maybe not necessarily physical characteristics.
Well, I think that the approach can happen both while the female is perched and while she's in the air. But the main aspect of the female
tell us about diversity of odonates here in Monterrey.
So to repeat again, I'm going to be talking about the diversity of odonates, which is dragonflies and lionflies in Monterrey. And to start off, we have learned in our classes that there's almost 1 million different species of insects. You can see it right over here, that there is more insects than there is other animals and plants combined. And there are estimates that there's up to almost 8 million undiscovered species yet to find. So they make up the bulk of terrestrial food webs. That is to say, they are mostly what gets eaten and they are mostly what eats things. And that helps keep an ecosystem interconnected. The more links there are, the more stable it is to interactions. And so if you look at this table or this graph over here, we have species of plants on this vertical axis and altitude here on the horizontal axis. And it's showing for each species the space that it takes up in the habitat or ecosystem. And you can see how the majority of them fall in this middle area. This is showing us the mid-domain effect, where there's more species found in the middle of the habitat. However, global warming caused by climate change is it shows a possibility of affecting this, because different species are being affected differently by global warming and may move to cooler and higher altitudes. One example is the Aedes aegypti mosquito, which transmits dengue, yellow fever, Zika, and chikungunya. A study done in Mexico in 2012 actually found that the elevation increased 300 meters. And that was exposing many different people to this mosquito, which they had never been exposed to before. And if you can think in human concept of disease and us being affected by a mosquito, you can also think of other species being probably harmed by this, outcompeted, etc. So global warming and rate shift shows a lot of potential to interrupt our ecosystems. And now we've heard a lot about Odonata already, but I'm still going to go through it again to make sure we're all on the same page. Odonata is an order with 2 suborders. One of them is damselflies over here. You can see they're a lot skinnier, and whenever they perch or they stand on a branch, their wings are like this. And then with dragonflies, or Anisoptera on the right, they will have their wings like this, open, and they're also a lot thicker. So there's a visual difference between them. And there's over 100 species of odonates just in Monteverde itself, and they're very important for the stability of our ecosystems. One way that they're important is that they're a trophic intermediary. So like I said before, they eat and they get eaten. They eat a lot of flies, midges, mosquitoes, and they're really helpful for pest control of mosquitoes. They also get eaten by birds, lizards, frogs, spiders, even by other dragonflies and damselflies. So they really are important for the transfer of energy in many ways. The second way that they show a lot of importance to our ecosystems is because they link aquatic and terrestrial habitats. In other words, they're a habitat intermediary. So they'll start off being born in water. They'll live their lives as nymphs, and they're actually predatory nymphs, so they will eat other insects in the water before going through a metamorphosis, getting their wings, and becoming terrestrial and then hunting in the air. So, we don't know much about a lot of these genuses, where they're at, their distribution and their range, what species exist. So my project was mainly to get a baseline of information to compare to in future studies. So I looked at how the diversity of odonates in Monteverde, Costa Rica changes in different elevations. So I had 4 study sites at 3 elevations. The lowest was the San Luis River, and it was at about 100 meters in elevation. And then it goes up to 1,100, sorry. 1,100. And then there's like a 100-meter difference, and we're at San Carlos Station, and then a 100-meter difference, and we're here. These are both at the same elevation, and here's some pictures of the sites. And it was a really fun project because I just carried around a GPS, I got my butterfly net, and I took pictures of the dragonflies and damselflies. There's one there. I tried to sample on sunny days because that is when they're most active. Odonates are ectothermic, so they get their energy and their metabolism based on the heat that they receive from the sunlight. So more sun means more activity. And I tried to limit the time or effort spent looking for them to 2.5 hours per site. That didn't happen in some cases, but there was an attempt. And And then after I got pictures, I would try to identify them using this field guide by Colton and Haber. But as you can see, there's a lot of similarity. These are all different species within the same genus, the genus that Sophia was talking to us about. So then when I needed to, I talked to Eladio, the identification king. He went and helped me catch a lot of odonates one day. Also Caligula, thank you Caligula. Yeah, and then from looking at my data, I got a few graphs. This is one of them. And you can see here on the horizontal axis is the elevation, and on the vertical axis is a number of odonates, so this is dragonflies or damselflies. And you can see how there's a lot more catches at this mid-elevation site of San Gerardo. So we caught more odonates in San Gerardo. And here are the exact numbers of the catches. And this looks like a lot, I know it's scary. But to think back of the mid-domain effect, you can see that this site has more X's, more presence of certain taxa. And so this is just kind of a visual to see that there is more taxa found at that mid-elevation site. And something else I thought was interesting was that this dragonfly, Brechmoraga rapex, was actually not found here, which is something that I'll touch on later. I also did some calculations of diversity. So to refresh, Shannon index is just a measurement of diversity. The higher the number, that means the more diverse that area is. And over here, D1 is the same thing. The higher the number, the more diverse. D0 is the richness. So in this case, since I'm doing genus diversity, this is how many genera were present here. This one was very high. And then over here is just a count of the most common species. Like how many species are very common to find. So, like I said before, San Gerardo had the highest genera found of all 4 sites. These are the diversities. So if you look at them all, the highest diversity was consistently in San Gerardo. Lastly, very important, is that the effort was not consistent throughout the sites. So as I was saying before, San Gerardo had the highest diversity, the highest amount of genera, but also it had the highest amount of effort because I had 2 helpers with me, Eladio, which honestly I should have counted as 2 people, also myself and Caligula. Yeah, so that could skew some results. Oh yeah, and FJ helped me a bit too. So while San Gerardo may support the mid-domain effect because it has the highest number of odonates, genera, and diversity values, it also has the highest amount of effort extended, so the data can be skewed. That might not be very accurate. And this species actually has a known elevation from 700 to 1,700 meters, but I did not find it at our low elevation site, even though that site resembles their preference for like rocky streams and rivers covered by forest. So I think that would be really interesting to follow up on in future studies to really determine like, is it a sampling error, or did I not exert enough effort, or is it actually missing and is climate change pushing it towards higher elevations? So yeah, to summarize again, San Gerardo may support my mid-domain effect theory, and this species of dragonfly might have a shift in its distribution. All right, thank you everyone for all of your support and help, especially to the people who taught me so much about science in Costa Rica, to the people that helped me out with going out in the field or like editing my paper, to my family who I don't think is here, but I love them and I miss them, and my friends. Thank you very much.
There's plenty, enough time for questions.
Jamie, nice story. I like how you recognize that more effort in San Gerardo may have an effect. Besides that, do you have a given consideration, because San Gerardo is Atlantic slope and the other sites, except for maybe the continental divide, are more like Pacific slope. How can that could different species? For example, the one that you didn't find in San Luis,
Yes.
is it more Atlantic species?
Yeah, so this is the only Pacific one. And unfortunately, I don't have any mid-elevation site for to reference for just the Atlantic site because this is low elevation. These are both high elevation, so I couldn't make any definite conclusion this was like a last minute decision to sample there because it was mid elevation and I did want to at least have that elevational gradient of low, medium, high. I think that's also something I should consider in the future if I want to study this because that would give us a clearer picture.
Yes, and maybe looking for the known distribution of the species between the slopes could help you to evaluate.
Thank you.
Very good.
Did you notice any differences in the environments that you were capturing a lot of the odonates in? So like, for example, if there's like sunspots just in the forest versus like you were capturing a lot in the river.
Yeah, so that's a very good point as well. I tried to keep it consistent and get like a relatively consistent stream of water at all sites. At San Gerardo, there was actually less water, which I found interesting. It was also a very sunny day. I tried to keep all day sunny, but that was a very sunny day as well. I was sweating a lot, and so usually with more sun and more heat, there's more activity. So that could also be a confounding factor. Yeah.
Were you only factoring in dragonflies and flies that you were able to catch, or were you also able to just identify them without catching?
Yeah, there's actually a couple that I have a much larger count than shown in the first graph. Because there were some that we got pictures of and we were able to identify later. And we took some great pictures too, a really big red one if you guys ever want to see. I can show that to you because there's some with very distinct marks that would be easy to identify that way. Yeah.
I'm really intrigued by the pattern actually within sites where there seems to be an increase in captures within sites. Do you have any explanation for that?
Yeah, this one was definitely because I got help from two people. So I spent, I think, about 2 hours here, but I had two people helping me catch at the same time. So it's like if we had spent 6 hours trying to get dragonflies and damselflies. And so that's probably why there's more catches there.
So I mean actually within sites, so like
Oh, here.
for San Luis River, there's more captures. Is that the like
So this is
around 1100 compared to
yeah.
a little like
Oh, with it being split up like this.
Yeah.
Oh, so I would walk along the trail along the river, and so there'd be some slight elevation changes due to that. Did that answer your question, or am I misunderstanding?
Seems like that way.
Sí.
Una pequeñita aclaración. Después de la tormenta Ney ha habido una disminución muy significativa en las especies. Y otra de las cosas es que en San Gerardo fuimos a ese lugar porque yo conozco el lugar y sé la cantidad de, bueno, prácticamente sabía que habían varias especies ahí, porque yo conozco el lugar. En este lado Pacífico sucede lo mismo. Nosotros, por ejemplo, cuando yo trabajé muchos años en esto y había muchos lugares estratégicos que ya los conocemos y sabemos que si va a esos sitios va a encontrar una cantidad muy significativa de especies ahí. Entonces, es como, depende de dónde vaya.
Sí.
So just to clarify that after the storm was complete, then we had this decrease in the amount of species in there. And also clarify that it's not Gerardo, this place that was chosen was specifically because he knows the place, knows the amount of species that are around there. Same with the areas chosen on the Pacific, so that also affects like the specific areas that were chosen because of the things that he already knew about the species and quantities.
That's good advice.
thought the presentations were awesome. And now we're going to learn about potential use of gavilana as insect repellents by Elizabeth Barrio.
Hi, my name is Elizabeth. I go to UCSD and I study the effectiveness of gavilana, which is a medicinal herb in Costa Rica. It's very common here. It's actually, when I was talking to my homestay mom about it and I told her I was looking for gavilana, she said she kills it because it's a weed. So it's very common here and I'm looking at it as an insect repellent. So, introduction, gavilana is native to Costa Rica. Its distribution is from southern Mexico all the way down to South America. It's most commonly used for stomach viruses, but like a throwaway secondary comment in a lot of the medicinal herb books that I read said that it could also be used as insect repellent, and that was really interesting to me because we've been getting bit by bugs since day one. So, I wanted to look and explore that because nothing works, no insect repellent works here. So, after doing some research, I looked up some research papers that talked about the chemical compounds that might be in it that help aid with the medicinal properties. So specifically, nerolin B and lobatin A, these are what are aiding in the insect repellent properties, but there's also nerolin A and lobatin B, which are in it and they help with these stomach viruses. So it's kind of like a little combination of both. The commonality between all these compounds is that they have a very similar chemical structure, which is known to exhibit medicinal properties. So you can find this chemical structure, like similarity, in other medicinal plants and other compounds made in labs, and that's really common, is that they try to copy this chemical structure when they're making the medicine in labs. So in one of the research papers I looked at, they were looking at these two compounds, nerolin B and lobatin A, as feeding deterrents. And so, like, looking at if an insect was willing to eat it and seeing how the insect reacted to it. And one of the things they kind of threw it in their discussion, like, they were curious about it, but it wasn't part of their main research, was a topical treatment, putting nerolin B directly on bugs, how that affects the bug. So the most outstanding reaction was that a large milkweed bug was paralyzed in its hind legs after being treated with this nerolin B. Until its next mole, it was fine after that. But that's what happened to the bug, so it kind of introduced this idea of like, how is this interaction, what's happening, like, you know, what's going on. So that introduces my question, which is, how does effectiveness against herbivory change with varying concentrations of gavilana infusions? Here, herbivory is in the question, but I'm not studying herbivory, I'm using it as a tool to study, like, insect deterrence. So, yeah, that's the main topic. Methods, how I approached this was, I first collected herbivores. So these are wasp larvae that I saw in my original prelim observations. And originally this was not my question, so I kind of took a picture and left. And then I went back a couple days later and walked through the forest until I found them, and they were in the same spot. So that was good. So these are wasp larvae. They are from the Symphida group. They are an unknown genus, and I know this because we asked Kenji, this is Emmy asking Kenji what it is, and he said that that's what it was, this was the wasp larvae, and I think Emmy gave him my last, ones that were living. The ones that were living after the experiment, she gave them to him because he was studying them. So that's the first one, and then the second one I studied, which Emmy found later in San Luis, were grasshopper nymphs from the Chromicus genus. And this was ideal to have two different orders, two different plant treatments, because it kind of shows that if there is an effect of gavilana, it is universal. Obviously, I haven't studied all the orders, but you know, there's two. Okay, so the way that I made the infusions, this is a gavilana plant. It is right behind this building. It's like over back there in the institute. And there is 4 beakers that are each treatments and 1 control, which this was just water, so this was like an untreated control piece. So the first one was the low concentration. If you can see it back down here, it's 0.006 grams per milliliter. The medium one was 0.024 grams per milliliter, and then the high was 0.098 grams per milliliter. So the way that I went about making these was I would pick leaves and then I would grind it up and I would weigh them so that I had a kind of I had a way to calculate concentration. Then I would change the amount of water to have it more diluted. So for the low concentration, there's 314 milliliters of water, which is a little over a cup. Then for This medium concentration, 78.86 milliliters of water, and then the high concentration is the same 78.86 milliliters of water, but there is about, 6 grams more of gavilana. So that kind of altered the concentration was the amount of, plant to water ratio. Okay, here are my methods. So this is what a trial would look like. This is the wasp larva. I would starve them an hour before they were put into the petri dish, just to prompt them to eat, because if they're hungry, they'll eat, So this is an example of the 4 treatments. Say this would be the control, so it's dipped in water. This is treatment 1, it'd be dipped in the low concentration, for example. Treatment 2 would be dipped in the medium, and then treatment 3 dipped in the high. And then these tape pieces behind would be labeled so that I knew what each leaf had on it, what treatment it had. So then when I decided to start running it, this is what it would look like. It would have a mesh over the top to allow airflow so that there's no mixing of scents or anything like that, and that the bug wouldn't escape. And this is what it looks like at the end. These are 2 examples of what it looks like. As you can see, there was an obvious choice for herbivory here, and then same thing here. This is what it looked like. And then using the Leaf Bite app, I would take a picture of each leaf and you kind of draw around where the original leaf was, and it tells you the percentage of herbivory done on those leaves. So results, here's what we're looking at. So this is the average of herbivory across all 13 trials I ran. This is both wasp and grasshopper data combined. As you can see, there's a strong trend going from the lowest concentration has the most amount of herbivory, going down to the highest concentration has the least amount of herbivory, and then this is our control. So yeah, these letters basically mean that they're statistically, if the letters are the same, so for example, our low and control group, they're not statistically significant, they're not different enough in herbivory data to be statistically significant. Versus our A and B groups, their data was different enough to be statistically significant. And then looking at, this is just the wasp larvae data, so this is only the data I got from wasp trials. And same thing, you can still see a strong trend going from the lowest, has the most herbivory, down to the highest, having the lowest amount of herbivory. And then the grasshopper, this one is not as obvious, but it's still there. This is 14.91%, this is 14.40%, but there still is a trend going down. Okay, discussion. So basically, the gavilana treatments with higher concentration of plant matter to water showed a low amount of herbivory. This is basically suggesting that the, going back to our intro, we had Nerlin A, Nerlin B, Lobatin A, Lobatin B, and medicinal properties. As the amount of gavilana increases, so does the Lobatin A and Nerlin B, which are our insect deterrents, so that also increases with it. So then we see also a decrease in herbivory because that is the effect that it's perpetuating onto the insects. So yeah, the effects get more obvious as the amount increases of gavilana. Then our low concentration treatment, if you remember that back to the averages of all, was actually the most fed on over the control piece, which was really interesting to me because the control was what they originally eat. But two things that could explain this is that there's a leaf part preference. So I would cut the pieces of the leaves in little 2 by 2 centimeters, like squares. And so sometimes some leaves had veins left in them, or part of the stem, or some didn't have either of those. And in a research paper I had read, it said that 2 factors go into where an insect decides to eat. So it's a combination of how much force it's going to take to bite into that leaf, and how many nutrients there are. So if there are more nutrients, for example, in the low concentration treatment piece, it might choose to eat it anyways because the amount of gavilana that's in it might not be enough of a deterrent to, dissuade it from eating that piece. So then that's what it explains here is that basically the properties in gavilana are not enough to irritate the gustatory receptors. They're kind of there's these GABA receptors that I had read in another research paper that basically said that is what's interacting with the Nerlin B is the GABA receptors, and it creates an irritating feeling. So if the concentration was low enough that it does not create that irritant, then they'll still eat it if it has the most nutrients and it's easiest to eat. Okay, the last piece is that the high concentration treatments consistently have the lowest percentage of herbivory. So like I mentioned earlier, this is a result of the interaction between the, basically the taste buds in the insect's mouths and the medicinal properties, Nerlin B and Lobatin A. These properties basically are irritating enough that they don't want to eat those leaves. And that's all. Acknowledgements, thank you, Emmy. You guided me the entire time, and you found my grasshoppers when I was in the hospital. So that was nice. And then thank you, Fede, for answering your phone really fast when I texted you. Thank you, Kenji, for answering the question that I asked him. And Brent and Institute for allowing me to use your lab and the station. Thank you.
There's enough time for questions.
Yeah.
Did you read anything about like applying Gavilana on the skin, like if it has any types of reaction?
No, I did not see anything about that when I was doing literary research, so I'm not sure. But that's kind of like the tool that I was using herbivory for, I guess, was to eventually see if it could be used as an insect repellent, seeing as the bugs obviously don't like it. Right. Any other questions? Yes?
How did you decide on the different concentrations for each group?
Okay, so yeah, so the the medium concentration, basically that came from a book that was in the EAP library where it said like 2 leaves for a cup of water is the normal amount for a tea infusion. So that's the concentration of that amount. And then to make the low and high, I just tripled it and then diluted it by 3. Emma?
What made you choose those specific herbivores to study?
I just found them. Like they're the ones I found.
It's interesting, calling the attention that the interest is to avoid mosquitoes and things. You are testing things eating plants. So is there a way for combine or can you apply these results for avoidance of mosquitoes or biting things?
Yeah, so, you know, if like future experiments were to see if Gavilana has an effect or does not have an effect on skin, and you could put it into an insect repellent, I think that because I know that the interaction is between the, you know, the taste buds of a bug and the Gavilana, that if a mosquito were to come and like interact with it, they would leave because they don't like that feeling. So I feel like that could be applied, which is, you know, that's what I was looking into originally.
It's interesting because also you have the search of the compounds that you are trying to see the effect on the insect. Nice.
Yeah.
Do you know if like Gavilana is like alright for human consumption? I mean, obviously like not straight, but like if there's any chemicals in it that would
No, it's most commonly used for humans for stomach viruses. They usually, like, they did exactly what I did in the lab and they make it a tea. So it's just leaves, they pour hot water over it and they drink it.
Okay.
So, yeah.
Thank you, Isabel.
It's a keynote presentation. It's in Keynote, not PowerPoint. But all you have to do is just click on it and open it.
You're bugging enthusiast. Guess what I do my projects on? Bombs. How'd you know? I did that with Dr. Brett. Yeah, I got it. Okay, 6 days. Okay, 6 days.
by Joshua Williams. We're going to learn about pollination trends in moths. That's the first.
Okay, well, Emily already introduced it, but yeah, basically an analysis of pollination dynamics. So for an introduction, basically plants really need pollinators and pollinators really need plants because insects are crucial for the health and productivity of all terrestrial life forms, and plants depend on insects for pollination because they need to produce fruits and seeds. However, currently there's a lot of regions witnessing a 10% decline in insects, which is possibly affecting their floral abundance. A lot of this is due to anthropogenic climate change, and something that's happening in Costa Rica, as we can see right now, is that Costa Rica's rainy season has increased by 2 months, which is affecting a lot of insects who rely on weather cues for germination. Many insects are, a lot of plant species have a great dependency on pollinators and therefore are more vulnerable to this pollinator decline than those who utilize other types of pollination such as wind and sunlight. Okay, so bees and adult Lepidoptera have adopted a total dependency on plants for food, and therefore have created structures and behaviors that facilitate this relationship. A distinctive characteristic of moths and butterflies it's a proboscis, which is this thing. And this is a long, extensible tube-like structure that's connected to the mouth parts and used for siphoning nectar. The proboscis unrolls with hydrostatic pressure and recoils when the individual is at rest. Lepidopterans have been around for around 190 million to 195 million years, and are probably the most underrated pollinators because most research is done on diurnal or daytime Lepidoptera butterflies. Lepidoptera are important for pollination for 3 reasons. One, because they display diurnal, or active during the day, crepuscular, or active during twilight periods, and nocturnal, active at night. Second, they transport pollen across large ranges of distances. And third, they are a very species-rich group and therefore have many pollinators. Moths specifically make up a majority of these pollinators, and there are 21 families that pollinate at least one family of plants. Of these 21, the most generalist are Spingidae, or hawk moths, which pollinate 35 plants, Noctuidae, which pollinate 24 plant families, Geometridae, which pollinate 18 plant families, which I used in my research, as you will see later, and Pyralidae, which pollinate 15 plant families. So my question was, do different species of Lepidoptera in Monteverde exhibit varying levels of speciation? I don't know. So for my methods, I took data from May 12th to May 17th of 2024 at 2 study sites, Bajo del Tigre and La Asociación Biológica. And I used light traps, which is basically a white sheet and 2 types of light, black light and white light, set up at around 5:30 p.m. Insects are disoriented by artificial lights because it affects their perception of orientation and therefore causes them to land, which facilitates my ability to collect them. To handle the moths, I held them by the forewing, as you can see here, the forewing veins, as to not disturb the scales that we all learned about in Emmy's lecture. I then held them off over a piece of clear tape and uncoiled the proboscis using a dissecting needle. That's the tape. That's a dissecting needle. It was really hard to do because they like to flail their legs around. As soon as I was done, I released the proboscis and they retracted it back into their mouth and I released them away from the light trap. So under a microscope at 400 times magnification. And I categorize them by morphotype or by physical attributes. And these are my 11. Oh, okay. One is missing. Oh! Okay, these are my 11. Alright. So a photo is taken of each so I can make a digital pollen library, which is kind of like this. And then my moths. I characterize them into 4 morphospecies using families and physical attributes. So there's Geometridae A, Pyrroloidea A, Pyrroloidea B, which looks very similar to Pyrroloidea A, but Pyrroloidea A, no! Has much more rounded, like forewing tips than Pyrroloidea B. And then Pyrroloidea C. So this is my table of 11 pollen morphotypes and my 4 morphospecies. So all morphospecies of moths carried at least 3 pollen morphotypes. Pollen 01 was found on all morphospecies of moth. Some morphotypes were only found on one morphospecies of moth, such as P06. And then 8 out of the 11 morphotypes of pollen were found on Geometridae A, except for P06, P08, and P10. On Geometridae A. On Pyrroloidea A, there were 3. Pyrroloidea B and C had 4. So, Geometridae A displayed the highest level in terms of visitation to the different plants of generalization. Pyrroloidea A displayed the lowest level of generalization. Pyrroloidea B and C displayed the same level of species specialization and shared 2 pollen morphotypes, P01 and P02. So this can be inferred that these moths are less vulnerable to floral decline because they're more generalist in their preferences. Some Geometridae are known to be specialists, such as Eos species, which are hypothesized to species that specialize on Piperaceae in the neotropics. And in turn, 5 out of 11 pollen morphotypes were found on multiple different moths, meaning that these plants, which create these pollen morphotypes, are most likely less vulnerable to insecticide. And my study was only a small fraction of moth pollination in Monteverde because I only had 2 families and my sample sizes were very small. So for the future, I would include more moth families, and I would also try to look more into what specific plant species are creating these pollen morphotypes. In conclusion, all moth morphospecies are generalists in this study. They all display different levels of specialization, and this may be due to pollinating only one plant. So thanks to Emmy, my girl, and Frank for letting me use his electricity, and Frederick for driving me into the Mega Super, and just all over the place. And then Carmen and Marvin for the Estación Biológica, and then Vita and Lorenzo, my homestay parents, for letting me use their house originally, but it didn't work out.
There's enough time for questions.
I have 2 questions. One was like, how did you pick the species? Like, who was the one who caught the most? And then also, like, how exactly did you get the pollen like from the boxes under the sea?
So for the species, it was just basically whatever I could catch, and then at the end I categorized them into different morphospecies. But at first I was looking at a paper done by Jonathan Coop in 1993 of Sphingidae moths, who are like much bigger and much easier to like pull the proboscis out of because they're like around this big. But I wanted to do something different, and I couldn't find any stingray, probably because of the season. And then for the collection, do you just mean like how, like?
Like, did you like how did it get onto the tape?
Oh, well, basically, like I was holding it like this. Sometimes I'd have to hold them upside down because they kind of like flail their legs, and then they get stuck to the tape. So I kind of would put it on like the side, an edge of a table or something, and then try to place them like at the edge of the table and unroll the proboscis so that like their scales and everything wouldn't get on. But that wasn't always possible.
Did you try the ID the pollen that you saw in the microscope?
No, I cannot do that.
There's a go-go.
Like, I think I could do it if I had more than 2 weeks. But definitely not in this study.
Yeah, because I was going to ask the one that you showed that only appeared in one of the morpholess species, I wonder if like they have some sort of, I don't know, mutualistic relationship where it's like, specific to that.
Wait, which?
The one at the table and then you showed that like the P something we showed in one morpholess species, like the P6.
Oh, this one?
Yeah, like do you have any degrees?
I mean, it could just be that it's more specialized to Piroidea, but I would need a much bigger sample size to know that. Hey.
Thank you. Sorry if you said this and I am lost. So these are the morpho species of moths that you cut, but is there a given idea about how many individuals of these morphos? I mean, is there a way that maybe geometry A is very common to catch and you have more samples?
Oh,
how even are those samples?
They are not very even, and that is a big error in my study. But yeah, basically in total I caught around 36 different moth individuals. And then the only ones that were exactly the same were probably, oh my god. I had a lot of these, so I probably had about 6 of these ones. And then Piroidea A, I only had about 2. Piroidea B, I had about 3 or 4. And then Piroidea C, I had like 5. Yeah, so it was very unbalanced.
Thank you.
How old was your study?
May 12th to May 17th.
Oh wow. How did you catch? Sorry if you said it.
Huh?
How many did you catch?
Oh, 36. Okay. That's it.
with perfume flower plants like orchids, where they are attracted to the scents and they kind of go and they like will sit on it and like rub the scent and put it in their hind legs and collect just tons of scents throughout their life. And they're known to be generalists, so they feed from various types of plants, and they're found in the lowlands up to 2,600 meters. So they got a good range. So my central question, let's first start with the gap in the knowledge, not to get too ahead. So gap in knowledge, there's been many studies on local abundance and diversity of euglissines in Monteverde. A lot of past EAP students, so I'm like, following a long lineage or whatever. There's other studies that also focus on species scent preferences, but there's very little known about visitation patterns in terms of pollination. So my research questions are, A, are visitation trends within euglycine species constant across diverse habitats? And B, do individual bees of a species show differing trends? So now my methods. What was I up to? So I had 3 different study sites. Monteverde, here at the Institute, literally just back there. And I was there for 3 days, approximately 4 hours per day. Then San Luis, which was like kind of near CIE campus, which was funny. Our rivals. And I was there for 2 days and spent less time there.
And then lastly, in San Gerardo, when we were there, everybody was there, and you guys, well, half the group was there. And you guys saw me collecting bees in the morning on 2 separate days. So my procedure, catch, pat, mark, release. Really simple, right? So catch the bee. Here is my line. I had a plastic line, and I had 3 napkins with 3 different scents that are known attractants. So I had eucalyptus oil, clove oil, and vanilla. Best smelling methods, I'm sure. I mean, some people were at the beach with like crab poop. It smelled really good. Okay, and then I would set them up in the morning on like sunny days when it wasn't raining, and the bees would come. I'd literally wait like 5-10 minutes and they were there. And then using an insect net, would go and like SpongeBob catch them. And then I would pat them, so put them into a glass vial. I would transfer from the net over, and I wouldn't touch them with my fingers so that I wasn't like contaminating them or like moving pollen around. And then put them into the vial, and then with forceps and like a tube of tape, like how you like that, you know, where the poster is like hung up on the wall, it's like that. And then I just hold it, and then I just tap the bee all over. On the underside, which is where like their like proboscis sits, so I got pollen from all around. And then before I released them, I would mark them. So here's an example of 3 little green dots that I put on the sputum, which is like the upper back of a bee, and then I would release them. So there's a cute one. You can't really see, but it's smiling. And that was so that if I were to recapture it later, I would know who is what, and I can compare examples. So here I have like 4 different genera that I captured. So here are 2 examples of the genus Euglosса, and then the genus Euphrasia, which is beautiful. You guys can't tell, it's like purple, green, blue. It's like insanely beautiful. And then this is Ulema, which is really, really big, really loud, and scary. And then this is Ixere, which is even more loud and even more scary, but very beautiful and just amazing subjects. Okay, discussion results. So, my overall findings. So I caught about, it was like 64 bees, and I found 50 plus unique pollen types across the 7 species of euglossines, which span, that's a mistake, 4 different genera. And then I found fungal spores as well as several mite species on my samples. And on average, there are about 12 different pollen types per bee, and in total per species, about 30 different types. And here is like an example of kind of like a Venn diagram that I made for each species. This is just Euglossa deceptrix. There were in total 40 different, over 40 different pollen types, and 15 were unique to Monteverde, 3 were unique to San Luis, and 2 were unique to San Gerardo. But you can see there's a large majority, 16, were shared between all 3 locations. And this is an example of a consistent visitation trend between species across all 3 locations. And this is the opposite, where there are regional differences in visitation trends, and you'll see when the numbers pop up. This is Euglossa imperialis. Same thing, over 40 different pollen types, but there are way fewer that are shared between all locations, and many more in the Monteverde and San Luis. They're just unique to those locations. So overall, bees of a species share about only 21.74% of pollen types across all 3 locations, so very few. So they tend to have regional differences in visitation patterns. However, it is like on a species basis, since you can see between the 2 examples, they are very different. And then here is a slideshow. I took way too long to do this. Like a lot of different pollen types that I found. So, I mean, there's also some that aren't pollen, which I found out later. But anyways, mites. This is, where actually is it? No, this one is a poaceae. There's a lot of poaceae, which is like grasses, which I think makes sense because they're very commonly found. This is actually a fungal spore, another kind of mite. These are fern spores. That one is my favorite. It looks and yeah, so you can see how like they differ a lot. This has like 3 little petals, this one has 6, this one has 3, but it's like a triangle. There's brown ones, there's yellow ones, there's like clear ones, tons of different kinds that I found. And okay, my biggest thanks to Annie for helping me and for like revising my methods in my paper, Frank for being around, for letting me be back there catching bees and like being wonderful and opening the lab for me too. I'd be like, hey guys, like at really random hours. For the Center of the Biological Station for letting me put my scents out and stuff. And then for all my friends who were there with me, helping me catch bees and keep it company. And of course, the bee between like Ulema and Glossa, so they're really interesting.
After this study, what further questions do you have? You have so many pollen.
Yeah, tons of pollen types. I definitely want to look into the like different resources that they visited, as well as like where the fungal spores came from, because I do know that some male bees, they collect pollen and they also collect scents, and some of the scents are like scatol, which is like a compound in feces, and like of course like when feces is like decaying, there's like, and obviously when a lot of things decay, there's like mushrooms and stuff, which is why there were like probably some interesting fungal spores. So I want to look into like what kind of fungal spores and what kind of like animal feces they're like more like attracted to. So like poop studies.
Oh, great.
Yeah.
Studies.
Sorry, I have one more quick question.
Did you notice that the bees tended to go to one like scent of base more?
Yeah, actually. So the majority of bees, they would go towards the clove scent, but it did kind of differ like on species and also on genus. So there were very few that went to the vanilla. It was just like Ulema and maybe a few Ixere. But the majority in Euglossa, because I got probably like half of my samples from Euglossines, they were mainly attracted to the clove and the eucalyptus. So I got some good photos. Yeah.
Earlier on in your project, I remember you saying that you were looking at the microscope and you saw human pee on one of the bees. Do you remember?
Yeah.
You said like large peeing on bees.
Okay, so I think it was Emmy who like reverse like Google image searched like one of my pollen things, and she was like, all I'm getting is like urine there, like infection stuff. And so I was like, who's peeing on my bees? Like, what's the point? I definitely did not look really too much into that because I was like, I don't want to know if this is true. But they were similar to a lot of the other Poaceae pollen types that I found, so I'm assuming it might be similar. Yeah.
I have two questions. Among the bees that you patched, you got different genera. Did you notice variation? I know you didn't measure this, but did you notice variation in the length of the tongue?
In the length of the tongue? Oh yeah. So okay, I tried my best to identify each sample that I got down to the species level. So a major factor in determining one species from another is actually the length of the tongue. I believe Euglossa, the Septipix, and Impedialis are like really similar, but the only difference is that Impedialis has like a way longer tongue. It's longer than the body. But yeah, it's really interesting because like even though these bees can be really small, their tongue can be like half or even double their size.
So maybe they are going to different types of flowers and stuff. Which, my second question, these are known as orchid bees, but sounds that they are going for perfumes, but not for pollen on orchid. Do you know? Did you find orchid pollen?
Yeah, so I caught 2 bees. That was like my closing thing on the question thing. Okay, you can't really tell, but there's like a tiny little orchid pollinia on the back of this bee. I only was able to catch 2 bees with pollinia. I guess it's just very uncommon. And a lot of other studies, and based on like Rubik and Hansen's guide, I also realized that it's just not that common to find orchid bees with the pollinia still attached. I'm assuming just because the pollinia is not like that well attached, but it can be like all over the body, and all different kinds of like orchids put their pollen in different spots. So like an orchid can have pollinators, like all kinds of pollinators, and pollinators can go to all kinds of orchids.
Thank you.
Thank you, ma'am. Okay, sadly, this is the end of this session. But don't worry, we're going to learn a little bit more about insect magnolia in the following session. But we're going to take a break in between.
Folks, in the next section, make sure that your presentation is on the computer, okay?
No, that was so good.
With us.
Oh yeah.
Guys, and then you're gonna learn it's all cereal card.
Well, what karaoke night?
Yeah, it's karaoke.
Oh.
Are we likely to get a bus back to the station?
Yes.
Yes!
He said,
No, we said we're rats, and then we should walk.
we're rats.
Remember how he says only rats are outside?
Well, that was my sister's and I say that there is
That's my answer.
Welcome to the fourth session about applied biology. Just as a reminder to speakers, it helps you, if once in a while you look over this phone, it'll try to get your attention and tell you that you have 5 minutes left, and 2 minutes left, and no minutes left of your 9 minutes, okay? The other thing is, if you have your stuff on a computer you're going to plug in, just come up after the person finishes. And then we have good news and bad news.
The same one, or is it
Good
two
one.
different?
The bad news is that Irene, who is the social media and visual arts person here at the Institute, wants to take a group picture. And I want to do it out in the rain. She says no, it should be under the roof at Fox Maple. So after closing remarks, which will only take a couple hours, we'll do the group picture, and then this is the good news.
We're busing.
So we are busing, yes, thanks to Naomi. I said, bad more. She said, no, the good news is that there are 2 more refrigerios, including donuts.
No!
You're lying.
I'm not, and including little pastries.
What? It's so much.
I know.
You never feed us this
Here we go. Nudia is going to be talking about water quality, and here we go for the fourth and last session. Thank
you guys. Alright, so today I'm going to be talking about the effects of tourism and urbanization on water quality in Monteverde. So to start, environmental concerns are starting to arise now that there is booming eco-tourism and more infrastructure being built. This can cause an effect on the health and safety of natural life. Our actions have a direct impact into water quality due to our contamination through pollution that disturbs all these aquatic ecosystems and the livelihood of organisms. And then last, our sources of pollution mainly come from roads, urban zones, and agriculture. And pollutants can include oil, heavy metals, chemicals, and litter. And a lot of agricultural practices also use pesticides and artificial fertilizers, which can cause eutrophication in many bodies of water nearby, which can pose a threat again to aquatic life. So for my central question, it's
kind of long, but how does tourism and urbanization vary water quality parameters including pH, temperature, conductivity, TDS, salinity, and macroinvertebrates among different streams in Monteverde? And before we start, I want to clarify about what all these parameters are. So pH is how acidic the water is. 0 to 7 is acidic, 7 is neutral, and then 7 above is basic. Temperature is important to look at because a lot of organisms can't survive at extreme temperatures. And then conductivity is how well a water can conduct electricity by indicating dissolved salts and minerals that are present. And then TDS stands for total dissolved solids. And this just tells us how many organic and inorganic matter is within the water. Salinity is how salty the water is. Again, there's limitations to this. A lot of organisms can't survive very salty waters. And then lastly, macroinvertebrates are small spineless animals that are used as indicators for water quality due to their sensitivity to pollution. And then these are my observed sites that I went to. So the ones in green are my urban sites that are highly impacted by urbanization and tourism. And then my blue is my rural sites served as controls for this study. And as you can see, those are a lot farther from a lot of developments and urban expansion. So they should have cleaner water. For my methods, I use an ED stick as my water quality meter to measure all 5 parameters. And then I use distilled water to clean the probe before and after use. And then for macroinvertebrate collection, I used a strainer and collected it for about 5 minutes to find as many as I could. And then I identified it with a bioindicator. Okay, now for results and discussion. So my mean value for temperature was higher at rural sites compared to the urban sites, demonstrating a statistical difference. And this can likely be due to sun exposure and elevation. Unfortunately, I did not take elevation into account before this project. But these two are in San Luis in the valley, so they are lower, which makes sense why they have slightly higher temperatures. And rural sites also tended to have a lot more sun exposure due to the fact that they were in open areas rather than the urban sites where there was a lot of infrastructure that was shading these areas. But nonetheless, the ideal range is 15 to 35 degrees Celsius, and all of these sites fall within that. Next for pH. The mean value of pH was slightly higher at rural, but we did not conclude a statistical difference, although the numbers are quite different. This is most likely due to calibration issues I was having, but the ideal range is 6.5 to 8.5, so all of these do fall within that range except for 2 sites, which is Catarata Murciélagos, which is slightly more acidic, and to this we can draw that back to the fact that a lot of visitors are visiting daily and they have lots of layers of sunscreen and lotion and bug spray that lower the acidity of the water. And then for Quebrada Maquina, it was a lot more basic than the average range, and this could most likely is due to the amount of less air pollution and less contamination in the area. But this is less of a concern because at higher pH levels, there's a decrease in the solubility of metals and pollutants, reducing their toxicity to aquatic life. Next, we have salinity. The mean value was higher at urban sites compared to the rural sites, showing a statistical difference. These values are due to, salinity levels should be below 1,000 ppm, so all of these are well below 1,000 ppm. And this is likely due to local geology and precipitation. So the local geology of all these sites had lush vegetation that most likely filters through all the unwanted materials that are causing lower water quality. And also precipitation can dilute the water so it's less salty and healthier for organisms. Then we have TDS. The mean value of TDS was higher at urban sites compared to the rural sites, showing a statistical difference. The ideal range is 100 to 500 ppm. So all of these fall sort of in this range. Again, this is probably explained due to personal care products that are being used at these sites. And also runoff at the urban sites would go directly into the urban sites, causing more dissolved solids within the water. And then the rural sites have a lot less visitors and are further away from developments, so therefore it makes sense why they have lower numbers. And then the last parameter is connectivity. For connectivity, the mean value was higher at urban sites compared to rural sites, showing statistical difference. The ideal range is 50 to 500 microseconds, and all of these do somewhat fall within this, except for Puricancha, the rural site. Puricancha is a reserve that likes to monitor and manage their water to keep ecosystems healthy, so it makes sense why their connectivity level was so low. While the urban sites suffer a lot, again, from runoff and from roads and fertilization contributing to this, to these higher numbers in the urban sites. All right, now for macroinvertebrates, there was a lot of water striders present in all of these sites, which is under the family Geridae. This isn't the best indicator; it's a neutral indicator because they can survive very highly polluted bodies of water, and they can survive very low polluted waters. So instead, I looked at the frequency of the other ones. So Nacoridae and Odonata on the rural sites are indicators of good quality. And as you can see, there's a lot higher number compared to over here, where Odonata is only one. And then also another interesting finding that I saw was that in Quebrada Sucia, I saw Chironomidae, which is a bad quality indicator. So it helped me identify, the quality, even though there was a lot more of neutral and positive quality indicators. In conclusion, all sites fall within regular to good water quality, just based off of the parameters and macroinvertebrates. Next, a lot of key parameters were affected. There was statistical difference in all parameters except for pH, again, likely due to calibration issues, but it's something to look at for the future. The need for management and monitoring is very important so that we can keep these numbers where they are so that they still remain in good quality classifications. And yeah. I'd like to thank Naomi as my primary advisor, Federico for helping me drive to all these different places and ask for permission, Emmy for identification of macroinvertebrates, and then Luisa for helping me confirm these identifications. And I'd also like to thank the workers that allowed me to go into Puricancha and Catarata Murciélagos. They allowed me to go even though you normally have to pay to get in, so it was very sweet of them. And that's all.
There's time for questions.
Yeah. So I noticed on the pH slide that like the waterfall, the Catarata Murciélagos, was like the pH was a lot lower compared to, was the Green Bridge like the one that you did right over like the Green Bridge right down the road from here?
Yes.
Okay. Because like, if I'm correct, I think those streams are like, those two areas are very, very close to each other.
Yeah.
Do you have any idea what might be causing that like pretty drastic difference in pH?
I think it's due to the fact that there's a lot more visitors at the Catarata Murciélagos. Like people go out of their way to pay to get in. So there's a lot more presence of people there rather than in the stream underneath the Green Bridge. It's just runoff from urbanization. Yeah.
I'm not sure if you got a chance to taste any of the water, but like, is there any difference in like the quality?
I did not taste it, but based off of my parameters, in my paper I was writing how actually all of these fall within potable water, so technically you could drink them. I did not try that, but it's possible.
I have a quick question. Do you have any impressions from just walking around how prevalent the release of gray water to the surface is?
No, I was not. I didn't specifically look at that, but the sites that were near roads, which is UCEAP, and then the stream under the Green Bridge, they are right next to like gutters. So like I'm sure that there is wastewater going into those, causing the differences in parameters, but I didn't specifically look at that.
So the Quebrada Máquina drains the station, and then also the Belmar and a bunch of other houses. Yeah, just gray water is a challenge.
Yeah.
Other questions? Thank you, Nadia. From UCLA, and she is going to be talking about different practices on the quality of chicken eggs.
This is my excellent project on qualifying the effects of varying agricultural practices on egg quality in Monteverde. To begin, the poultry industry is a global industry. Poultry includes both poultry for meat as well as egg production. And here in Costa Rica, 50% of the protein sources comes from the poultry industry. That ranges from multinationals such as Papasa or Walmart, which house like millions of chickens and are constantly producing, or the more prevalent backyard flocks that occur in like residencies or family farms. These can have as few as just a couple chickens. Alongside that, there are very agricultural styles here in Monteverde. We visited a few of them in our agro class. These include commercial farming, sustainable, regenerative, and family. While we learned about like dairy cows on some of these and plants on others, I was more curious about their implications on livestock as a whole and in specific poultry. And surprisingly, not all eggs are created equal. The only factor of an egg that is determined by the breed of chicken is going to be the external shell color. So I was really curious how these agricultural styles would affect other egg characteristics. This led me to my question: how does egg quality of poultry vary among different farms and their respective practices? For my methods, I did survey eggs from 5 different practices, one of them being a commercial and industrial production. And then the other 4 were smaller scale farms here in Monteverde. One is still commercial and is sold at the grocery stores, but on a smaller extent than Papasa. First I did external quality. This included visual inspection for like dirt or scratching and stains. It also included candling, which is another analyzation of shell integrity. It includes holding like a bright light up against the egg. It's a little hard to see. The top one over there indicates staining, and the bottom one is cracked. And then I also measured their weight with the electronic scale, and then width and length with the micrometer. I assessed internal quality next, which included albumin quality and yolk quality, albumin being the egg white. These were both measures of the diameter of spread relative to their height off of the table. And then I measured these with a ruler, both for diameter of spread and for the height. I used color qualifications to compare the shell color and the yolk color. Among my different samples, these are from an egg yolk quality evaluation site. And then simultaneously, I did perform an agricultural survey with 3 of the farms. The small-scale farms, this included Finca Margarita, Life Monteverde, and Granja Los Lirios. And this was kind of just to gauge the different agricultural practices on each farm, as well as in regards to poultry specifically. I'm going to talk about 3 of my results, even though I gathered a lot of different variables and parameters. The first one being shell color. Here in this graph, on the x-axis, it is separated by the different producers of the eggs, and on the y-axis, we have the average shell color. You can see that Finca Margarita has a lower average shell color relative to the other 4 producers, and that the standard deviation is also larger. Both of these results were statistically significant when compared to the other 4 producers. Just to kind of give you some imagery for what I'm even talking about. This one down here is going to be papasa, which is the commercial and industrial poultry production. You can see there's extreme consistency amongst all 15 of the eggs. They kind of all lie in one color spectrum. Up at the top we have Granchulos lirios. You can see they have the same kind of shades, a little bit more variation. And down at the bottom we have this one. We have Finca Margarita, and it's kind of all over the place in regards to shell color. It ranged from like a 2 to a 14 on the previous scales. However, in my intro I mentioned that shell color is a function of the breed of the chicken. And in my agricultural survey, I did find out that Finca Margarita houses 5 breeds of chickens, including Rhode Island Red, Naked Neck, and some of the other ones listed. At the bottom we have both Rhode Island Red and Naked Neck. Versus Life and Granchulos lirios reported only having 1 breed of chicken on their farms, which was the Easter Brown. And since shell color and breed of chicken do show a positive relationship, it makes sense why Finca Margarita, with their 5 different chicken breeds, would have had the most variability in their shell color. And then to explain papasa having very consistently looking shell color, this does correlate with consumer preference. Previous studies have shown that the inexperienced consumer does prefer a consistent look to their produce or their fresh products that they are buying because they correlate this with quality. Commercial poultry production such as papasa may be striving for that consistency as it is leading to more sales because people think it is quality related. Another index that I looked at was the yolk index. This classifies the quality of the yolk into 2 categories, and that's going to be high and low. This graph shows the farms on the x-axis and then the number of eggs within each of those classifications on the y. A high-quality yolk is going to be firm and tall-standing, as shown in the blue over there, versus a lower-quality yolk is going to be flat, squished, or more round. In these ones, you can see that, in this graph, you can see that low was more common amongst all of the farms. However, Finca Margarita did have a significant amount of high relative to all the others, and Avicola Marisol and Papasa kind of showed the same trend. Yolk index is going to be a function of freshness and the temperature of storage. Because structural integrity does decrease in the hotter the temperature the eggs are stored in and the amount of time they've had on the shelves or in their shipping containers. So this does again go back to the commercial farm versus family farm. A farm such as Finca Margarita, which we saw had the highest number of high yield quality, has an extremely small producer to consumer cycle. All of their eggs are used on their own property or within farms. So these eggs aren't sitting in like shipping trucks or sitting on the shelves in grocery stores and decreasing in quality as people aren't buying them or just time from the lay increases. Versus the commercial farms such as Papasa or Avicola Marisol, both which occur in grocery stores, did have lower freshness of their yolks, which makes more sense because they could have been on the shelf for a longer time when it came to me studying them or the potential time for consumption. The last result we're going to look at is the Howe unit index. This is an index that relates to egg white quality. There are 3 classifications. That's going to be high, moderate, and low. Same style of graph, farms on the x-axis, number of eggs in those classifications on the y-axis. This data is kind of, it did not prove statistically significant, which I'll get into. It's very kind of all over the place or similar. Up at the top, I will say that this index looks at the inner albumin, which is going to be the thicker one that you can see here on the closest to the yolk. Up at the top is going to be a high-quality yolk. You can see it's very distinct from the outer albumin versus at the bottom. The distinction between those 2 functions are blurred. This relates to the function of the egg white as a whole. The outer egg white is going to serve as a protective barrier, particularly against microbes for the egg, while the inner albumin provides nutrients for a potential embryo that would be growing in a fertilized egg. However, when laying hens do lay their eggs, the fertilization status is unknown to them until the time of incubation. So it would make sense that all eggs laid would have a higher inner egg white quality because it is a fitness advantage to provide the nutrients for their embryo. It would be a disservice to potential embryos to be laying eggs that don't have a thick and healthy inner egg white. And this trend was shown as my data did not not show significant difference amongst the farms, regardless of the practice and the size of practice, diet, whatnot. All of the egg whites were pretty consistent. Overall, like the kind of conclusion is that all of the parameters in which I collected can kind of be put into two categories. That's going to be conditional versus biological. Conditional meaning that the quality is a function of something that is relative to the farm. So whether that be the breed of chickens that the farm has, which can differ from farm to farm, or the feed that the farmers are providing their chickens, versus biological, those factors or parameters did not show statistically significant differences, such as egg white. This is something that applies to chickens as a whole, and so it makes sense that they're going to maximize that quality. My acknowledgements, thank you to you all, and any questions?
Questions? Yes.
Other than reducing the amount of time from the lay, are there any things that farmers could do to increase the quality of their eggs?
Some of the other factors that I unfortunately did not have time to go into are influenced by things like diet. The color of the egg yolk, for example, is determined by the diet of the hen and is more an indicator of hen health. So making sure that the hens are getting like a versatile, diverse diet and not just like giving them just purely bread or something. What was some other factors? But there are things that farmers can be doing, like the space that they're given. None of the small-scale farms that I surveyed give their chickens antibiotics unless it's absolutely necessary, so that could play a role as well.
This is kind of like a broad question. I don't really know if everybody's very on it, but do you think that the eggs that you looked at in Costa Rica have been of higher quality, like conventional eggs in the US?
I would probably say that they're similar to at least the industrial eggs that I surveyed, but definitely different in comparison to the smaller scale farms. I feel like I don't really know of any backyard flock type poultry productions in the States, and those ones did tend to have, on some respect, higher quality versus the industrial ones that are going to have like one standardized mode of care for their chickens, which could decrease quality. Yes.
It's interesting to listen to the studies. Hopefully we can apply to some. Caught my attention about the thickness of the cracker of the shell. Is there a, sorry if you mentioned it and I am lost, but is there a way also to characterize how strong is this shell or how thin it is? Do you know?
I'm sure that there is a way if there was like a more standardized way for me to break the eggs rather than just like using my hands and stuff to determine size or strength. I will say when I looked at the width of the shell amongst all of them, it wasn't statistically significant. But I did notice some had like I had a more difficult time opening them because they had like a, I don't know, protective layer of like.
Because I didn't get if the things that you observe are standardized methods or are the things that you are measuring to characterize this. So
it was kind of a mix. Like some of the things were standardized, like the Howe unit index, the yolk index, and I did use a lot of other previous papers on egg quality to make sure these were correct parameters for quality. Some of the other things I just gathered in case I would like to use it or it came out interesting, and I would rather have the data before I throw the eggs away or use them for other things than not have it.
Thank you.
And do you know if the physical traits are somehow correlated to nutritional traits for human consumption? Is one egg that is nicer and it's like really big, better to eat than the black one?
I do think a lot of the quality parameters are in regards to freshness, especially because egg yolk and egg white do deteriorate with time. They just lose structural integrity. I'm not entirely sure what the nutritional aspects are of some of the other ones. Like if I maybe looked at content like percentage of egg yolk versus percentage of egg white, that may have played a role in nutrition, but I unfortunately do not know.
Thank you.
You should have touched them and something broke them and broke them on the last one.
Our next speaker is Wex, and she's going to be talking about fungal biocontrol of ticks on cows.
Hey guys, I'm Wex, and this is my field trial of fungus, Metarhizium and Asophily as a method of tick reduction in cattle. So these are the cows at the Stuckey farm, and if a lot of you guys remember, we visited on one of our farm visits. And I was struck when talking to the main farmer, Sarah Stuckey, about how she's been having a big issue with ticks on her cattle. So, the problem, ticks, are obligate blood-sucking parasites that during their life cycle, they spend part of it in grass. So like in the US, you might go into the forest, or if you're playing around the grass, you might get a tick. Your parents might tell you to watch out for those guys. But also with cattle, in the pasture, they live on the grass as free-living organisms, and then for part of their life cycle, they latch onto their host to feed and grow and hold. And they cause a lot of issues for cattle and for farmers alike, decreasing milk yields, bringing diseases like cattle fever, and they've even killed some of Sarah's cattle. And so her farm and her goal is to transition away from chemicals to more organic methods, and so I thought, how could we do this? So currently, a lot of farmers globally have been using chemical archaicides, usually from baths or sprays, to deal with their ticks. About 80% of cattle worldwide are super susceptible to getting ticks. But there's a lot of issues with using chemicals. They can kill non-target organisms that are beneficial in the ecosystem. They contaminate water and soil. And the main issue that a lot of farmers are most concerned about is tick resistance. So they become resistant to these chemicals, which means farmers have to spray more frequently and more aggressively in order to deal with them. And here's some kissing babies, and then that's some of the chemical examples. So what are some other options? What are some biological things we can be using? So I did some research, thought about doing ducks for a second, but then I found this thing called entomopathogenic fungi. It's kind of a long word. I found a trial in Mexico where they were successful, and they used a strain called Metarhizium and Asophily. It's like the most common commercially and in studies. Essentially, it is a natural enemy of ticks, and scientists have been using it to infect and parasitize arthropods. So some of the benefits of it are it targets ticks at all their life stages, and there is a low resistance, which is the key for farmers. And this is the product, and that's an example of what they should look like when they've been fully colonized by the fungus. I'm going to go quick through this, it's kind of complicated, but essentially the conidia, or the spores from this product, they land on a host, suss it out, see if it's a good place to be, and then they germinate. So they'll come through here, through the outside of the tick, go through, and then eventually kill the tick using mechanical force and enzymes, take over the organs of the tick, and then fully colonize and eventually create more spores in this cycle. My question. Can entomopathogenic fungi, Metarhizium and Asophily, be used as a biocontrol for ticks in real farm conditions in Monteverde? And how do different concentrations of this fungi impact the effectiveness of bee control? So, study site, Sarah's farm. Here is a picture of all the paddocks. So she rotationally grazes her cattle, which is super interesting in its own. Look into that. And here's some interesting information about weather, humidity, and the UV, which are coming to play later. So, methods. I use two treatments, a high and low concentration of this product, using a recommendation from a local engineer who knows the product and has used it before. That's Roher. He works on the farm. He's awesome. We mixed the product in water and then poured it into this backpack sprayer, and he sprayed the sides of the cows. We did it at 4:30 in the morning to avoid high UV radiation and rain, because the fungus it lowers its effectiveness when it's exposed to those things. So they had about an hour to sit and rest, and then they went out into the field, and about 2-ish hours before the sun rose, and they're exposed to the sun. And we had a control as well, with not being sprayed at all. And then, quantifying the ticks. So the day before and then every day after I sprayed, I went into the milk room, which you can see down there. They have 3 cows come out at a time, and then I would just kind of go up with my little notebook, count the ticks, and categorize them into ranges, as in this chart. So it was kind of hard sometimes. Most of those are flies, little dots you're seeing, which made it a little bit difficult to tell what was what. My results. So the high concentration overall had a 14% decrease. The low concentration had a 11.8% decrease over the full time. Oh, this is the average tick category, and then this is the full 6-day period. And the control also had a 7% decrease over the full period. And you can see the trends are very similar, which had me wondering if they're all decreasing, what's making that happen, if it's not necessarily the fungi. Here we have some more data. So we have 2 tables, one for the first 34 hours and then for the full 6 days, because as you saw in the first graph, there's quite a bit of a decrease in the first chunk of the time I was looking. And all 3 decrease, but as you can see, so this is just looking at each treatment, and then if the amount of cows that decrease, increase, and stay consistent with the amount of ticks by percentage. So in both groups, the control decreased by around 50%, which was interesting, which implicates that something other than this fungus was at play in the situation. Don't know exactly what it was. Might have been when I was talking to the guys at the farm, saying that the ticks were at the end of their life cycle and they dropped off to lay eggs to replicate. It could have been potential contamination of other chemicals as well. So yeah, there's a lot of different possible reasons for this to have happened as well. And it's interesting to see also for the high concentration, there was actually an increase in the number of cows with an increase in ticks over the 6-day period, which kind of indicates that there was an element of randomness in the amount of ticks. So yeah, like when I first got there, we sprayed the ticks. About 34 hours later, we noticed a huge decrease, especially in the high group, but amongst all the 3 groups. And so that had me wondering, like, why could that be? I don't have a definite answer for that, and I think that would be an interesting thing to look at in a future study. So yeah. So here is an example, and it's kind of like jarring, but here is cow 8, called Susie, from the high concentration group. It may not necessarily be the fungus that was the reason for the decrease in ticks, but you can see there's a pretty steady decrease over the 6 days, starting with each one circled as a tick, like a mature, adult, engorged tick. And then by the end here, there's very few, there's a lot of flies, so I'm indicating that there. Just an interesting example of how it changed over the 6-day period. So, although so there was a high concentration treatment had the highest tick decrease, but also because the controls had a pretty significant decrease, it kind of means that the results were inconclusive. But I still have hope because there's a lot of ongoing research that's saying that this could be an effective strategy. So this is one study out of many. There are not a ton of studies that are actually in the field. Most of them are in laboratories, which is a lot easier to control for, and it's not real life. So that's something that I found to be interesting about this study. And here's the cows in there in the pasture. And then so farm feasibility. If this is to be something that farmers are looking to use, scientists are looking to use, the economic impact is something you can't ignore. So one bag of product that we bought was $27 US dollars for 500 grams of product. So that can make about 100 doses, about 27 cents per dose. There's not a ton of information in comparing like conventional chemical methods to this fungus, which is something that needs to be looked into more, and also there needs to be more commercialization. So it is available on the market, but not in the way that it is for commercial chemicals as well. And something else for future studies. So my time frame was about 6 days. A lot of other research uses much longer time frames because this fungus operates, it's a living being, it has a full life cycle, so it needs a longer time to be observed. So future studies, if I could do this again, have more time to be able to observe it. Weather conditions, as I said, the UV and rain come into effect, so it'd be interesting to study like exactly how that works. And then another factor is reinfestation. So, okay, I'll finish up. Reinfestation, so these cows are being cycled through, and so every time they went to a new paddock, they could get new infestations of ticks. And then, as I talked about before, not a ton of research on it, and it needs to be grown, and it's an exciting field to be working on. And I would like to thank Naomi for all her help. Could not have done it without her. And the taxi driver took me at 4 a.m. That was really nice. And of course, my cows. Thank you very much.
Time for a question? Oh, yeah.
Would you personally spray this spray on you if you had kids?
I think I would like to see some more definitive research on it working. And I did have it sprayed on me, actually, right in my face. When we were applying it, yeah, the machine, or like the backpack sprayer broke and sprayed me in the face. So, I lived.
What does it smell like? Or is it like sticky or what's it like?
I didn't do the spraying. It didn't smell like anything. It didn't smell like anything. It didn't smell like the Okay, is there not time? Or?
Yeah, that's it. Thank you. You have it on your computer, right? Karen, ¿usted puede bajar las luces a solicitud de FJ? No, un poquitito, no completamente oscuro. Es bajarlos, como apagarlos. Apagarlos. We're starting. Our next speaker is FJ Fiona Joy Wagner, and she will be talking about microbial diversity on the neuroskin.
Hi, my name is Fiona, or as you know me, FJ. I go to the University of California, Berkeley, go Bears. And today I'm going to tell you a little bit
drop it. Testing. Oh. Testing. Cool. Okay. So moving on. So this is a rufous-eyed brook tree frog, and it was captured in San Gerardo by Joe Zeno. So thank you for letting me use it in my slides. Here he is. Okay, so what are microbes? Can I get a volunteer from the crowd? You.
They're very, very tiny organisms.
Yes, they're microorganisms, but from what I heard from Naomi, they have a bad connotation in most general public, so you think like COVID and stuff. But basically, microorganisms are fungi, bacteria, and viruses. We've all heard about viruses, COVID-19. But basically, I did my study on some of the biodiversity in Costa Rica and their frogs. Anurans are frogs. Thank you, Janan, for explaining that earlier. Basically, we're in our 6th mass extinction, and yes, it's our fault. But I wanted to focus on frogs in that mass extinction. I think Janan also talked a little bit about the chytrid virus, aka the Batrachochytrium dendrobatidis. And basically, it was a huge problem in the 70s and 80s, where it spread over many, many, many frogs, basically almost all species, and wiped them out pretty seriously. So it's been a big problem, and we definitely need more research on fungi and other microorganisms that could cause future mass extinctions and even erasure of some species. And basically, I'm building a microbial library which showcases the diversity of some microorganisms. For the purposes of my study, I'm only focusing on bacteria and fungi. And a microbial library is basically a huge collection of different types and morphotypes of bacteria, fungi, or viruses, if we were focusing on viruses. So my central question is, what is the microbial diversity like on Anuran skin in Monteverde, Costa Rica? Okay, getting into the methods. We're going to start with the field portion. As you can see here, I have drawn a swab. You can see it, just like the COVID swabs that we used to put in our noses, if you remember that. And also the little bag and the label. Basically, I would go on like an hour to hour and 30-minute long night walks with Eladio Cruz and Janan, and we would go and catch frogs. The first time that I had ever caught a frog was pretty terrifying, if I'm honest, because they're kind of slippery and they jump around. And basically the way Eladio did it, I thought there was a methodical way. No, he just reaches out and snatches them from the ground, leaves litter and all. So that's what I did, fortunately, poor frogs. But I was trying to be gentle. So we take the swab and swab the ventral side, which is right here. Shout out BioNA if you took it. The ventral side and the leg and feet. I'd have 2 swabs actually, so this diagram is actually incorrect. And then also the dorsal side of the frog, and the leg and feet too, and the head too. So that just shows where I swabbed them, and I did it about 30 times, 15 times on the ventral side, and 15 times on the dorsal side. And put them in a little bag and sealed them forever. Okay, and the second part of my methods is the lab portion. I'm sure you've seen me in a lab coat, or a photo of me in a lab coat, or Joe and I in and out of the lab at the Institute. We've been busy. But basically, I take the 2 swabs that I took from each specimen of frog and save them either in the fridge or at room temperature. And I created, if you know what agar is, it's basically a media. It comes in a powder, and you boil it in water, and it becomes kind of a jello-like consistency after you pour it into like a petri dish or a plate. So we use potato and soy agar, and that's basically what microorganisms can grow on, and basically the potato was used to grow fungi, and then the soy was used to grow bacteria. So this is an example of a freshly, oh, that was labeled wrong. It's not streaked. Actually, it could be. A freshly streaked plate of agar, which is basically taking one of these swabs and moving it back and forth in a 4-quadrant pattern like this, so you can find individual colonies and see how they grow. That's the best way to showcase morphotypes from growing microorganisms. Yeah, so you can't see any growth here because it was just streaked. And then after 48 hours in the incubator, should look something like this. But morphotypes are pretty different for all bacteria and fungi, so you'll see a little bit later what it actually looked like. But yeah, and also the labeling here is potato, because this is a potato plate. Okay, so some results of the frog walks. We found 14, we were able to catch actually, 14 different individuals of frogs total within 8 hours of data collection, spread over multiple nights, and 7 different species of frogs. We have C. crassidigitus, D. rufiocoleus, P. ridens, C. fleshimani, C. bransfordi, L. foreri, and L. varsezitowichi, which is the last name. But the most species that we caught were clearly this one, so please make note of that. And also, the individuals per location, we found the most frog individuals in San Gerardo. Literally on just like one night, the first night out, we found 8 different frogs and swabbed all of them. And we had 4 different locations. So we had San Gerardo, the Estación Biológica, the Crandall Reserve behind here, and the Centurio Ecológico. And yes, as you can see, we found the most in San Gerardo. So we also have, we found them, I made note on what type of substrate we found each frog on. And so as you can see, we have the most species and individuals of frogs found on leaf litter and in the creek, and only 2 found on water plants, as you can see. And there was a lot of this guy in the creek. And then this was the morphotype library that I built. I'm not sure if you can see this super well, but we have this morphotype. You can see how it's growing like this, and I actually made descriptions in my paper, but for the purposes of this presentation, I'm just gonna tell you, like this bacteria or fungi was only found growing around the rim and was a light white kind of clearish color. These grew in spots. These grew in really small spots. This had a little green head, and they all looked pretty different. Yes, and here we go. As you can see, there's a lot of different types of microbes. And the thing is, we couldn't pinpoint if they were fungi or bacteria. Even though we did have the 2 different plate types, theoretically, that would have been enough to tell if it was fungi or bacteria growing on the plate. But because there's a lot of similarities in the potato plates and the soy plates, which would have been the differentiating factor if it was fungi or bacteria, didn't work out. So basically, we just built a bacterial and fungi library. If we had technology to work with DNA, then maybe we could identify that further. And so here are some of the microbe types and species. So the microbe type that had, was most common on all the species was microbe 1. As you can tell, it's like almost in every species except for this one. And microbe 5 was also pretty prevalent, while microbe 13 was only found on bransfordi, which is very relevant. And here's some more results from the substrate / microbes. So we had leaf litter, water plant, and creek, and because we had the most frogs found on leaf litter and creek, we also had the most microbes from leaf litter and creek rather than water plants. Yes, so Unfortunately, we were able to catch less individuals compared to previous studies. Even another spring study from like 2018, they caught like around 31 frogs, and they also went out for only like 9 hours, which was quite interesting because Janan could hear a lot of frogs, but we were only catching just a few of them. Some nights, even at the Symposium Ecologica, even Eladio was kind of surprised at the like less amount of frogs that were around the creek, because usually you'd find a lot of them there, but they must have all been hiding on top of the leaves in the canopy, so you could still hear them. But we were unfortunately unable to swab them, so that also led to less morphotypes being able to be found. And perhaps it was due to late rain in the wet season this year, or too much rain on the nights we went out, not entirely sure. But there was a positive correlation between substrate type and frog species. On leaf litter and creeks, they were the most popular, and perhaps that could be dependent on species preference, or if it's a wetter or drier night, they just would have been found on that. More research definitely needs to be done on that. And there was also a positive correlation between substrate type and frog species. There's a lot of conflicting evidence and papers found on if microbes on frog skin are dependent on the species, but it seems like in my study, which is a rather small sample size, it could be possible, but more studies definitely need to be done to determine that, the larger sample size. And perhaps there could be self-cleaning tendencies from frogs. I was unable to observe them in their natural habitat, like cleaning themselves, but it is said that they can like rub themselves a little bit or wash water over themselves, which could possibly wash away microbes and lead to less morphotypes that we found compared to previous studies. And also, either if we went out on rainier nights, which we definitely were in like thunderstorms, they could contribute to washing away many microbes on the skin or even adding them. But again, more research definitely needs to be done to figure that out and find more types of morphotypes that could be added to the library. And in conclusion, microbe type is positively correlated with substrate type, according to my study, and substrate type is positively correlated with species, but more research definitely needs to be done with larger sample sizes and DNA sequencing to build a much more comprehensive microbial library, and especially being able to determine if it was fungi or bacteria growing on the plate. And some acknowledgements, of course, Eladio. I wouldn't have been able to learn how to catch frogs without him. Fede for always organizing the taxis on time and checking in. Luisa, of course, for letting us mess around in her lab all the time. And thank you, Naomi, for always like encouraging me when I'm doing the study, even when I get really tired. And Joe, of course, the best lab buddy ever. And thank you so much, Frank, for pointing out that terrible spelling mistake I made on this slide set. If you can catch it, you win $10. And thank you, Emmy, for helping me with statistical tests. Thank you. Any questions?
One fast question.
What would I change?
Yeah.
Oh, I think I would request DNA technology so I could figure out if it was fungi or bacteria, or try to do more tests on soy and potato agar to see if there's a better determinant without the need of DNA testing. So, yeah.
know Edwin knows how to make that.
I do. Yeah, it's right here.
Wait, we were on the right page.
The next speaker is Joe Zeno, and he's going to be talking about microbes on hummingbird tongues related to foraging.
Hi, I'm Joe Zeno. I am a first year at the University of California, Davis. And for the last 3-ish weeks, I have studied microbial variation in hummingbirds based on their foraging habits. And as you can see here are the two foraging habits that I studied. Hummingbirds that feed from flowers, and hummingbirds that feed from human-made feeders. So, introduction. So, like I said, hummingbirds have two sort of sources of where they get their sugar from. They constantly are burning calories every day. They burn around 10,000 calories per day, and they need to be constantly feeding. So, two sources around here that hummingbirds feed from are floral nectaries from flowers and sugar water in artificial feeders. So, can anyone tell me what is wrong with one of these birds? So I need someone raise their hand. Lance, you got it?
The bottom right is all jacked up.
Yeah? Yeah, exactly. So, this green crown brilliant is a bird that is often feeding at the Monteverde Hummingbird Galleries. And as you can see, its beak is completely bent and its tongue is hanging out of the side. And even if you look really closely, you can see little black dots on the tongue. And this is a common pattern that I found with birds that were feeding from the feeders. This lesser violet ear is a healthy bird. You can see it has a pretty normal looking beak. But yeah, so basically this made me want to research what was causing this. And what I found was that microbes can live on the tongues of hummingbirds. And the microbes that I studied in this research consisted of fungi and bacteria. So my research question was, how do the health condition and microbial abundance of bacteria and fungi on the tongues differ between hummingbirds that primarily feed from flowers and hummingbirds that primarily feed from feeders? And my prediction was that the birds that feed from flowers will have healthier physical features and less bacteria and fungi on their tongues. So now let's get into the methods of how I conducted this study. First, I'll talk about where I conducted this study. The first site I researched at was the Estación Biológica, Monteverde. And the second place was the Kane property, along with another researcher, Deb, who was doing a bird banding study at the same place. And lastly, the Monteverde Hummingbird Gallery, where there were feeders. And so, how I conducted my field research was first by catching the birds with mist nets, similar to the mist nets described earlier in the bat projects. And then after that, I would collect physical data on the birds, such as their weight, the tongue condition, the beak condition, and feather condition. And after that, I would swab the tongues of the birds to collect microbes on a cotton swab sample. And then that cotton swab sample was brought to the lab. And in the lab, where I worked pretty much 100% of the time with FJ on the same sort of similar procedure was I first would cook agar, which is essentially a food for the bacteria and fungi to grow their colonies on in petri dishes. I would put the agar into different petri dishes. There were 54 total plates that I cultured. Then I would streak the plates with the agar on, and then I would incubate the plates for 24 hours. And this is the rankings. I came up with a ranking system to rank how the cultures grew. As you can see on the left, there were very little culture grown on this sample, and on the right there was a lot, so I ranked it from 1 to 5. And so here are my results. So for the beaks, there was not really any variation between the foraging sites, between the feeders and flowers. For the feathers, it was the same deal. But with the tongues, which was sort of what I predicted, there was significant variation in the physical quality of the tongue. And as you can see on this column graph, this is the average ranking of tongues of feeders and the average ranking of tongues of flowers. And I came up with another ranking system for this that ranged from 1 to 3, where 3 was in perfect condition, 2 was slight damage, and 1 was heavy damage. And there was only one bird that had heavy damage on the tongue that I caught, and it was at the feeders. And so here are some of the results for the microbes that I observed. At the feeders, there were more fungi on average than at the flowers, and for fungi, and then for bacteria, there was an even bigger discrepancy between the flowers and the feeders. And let's see. So now, if we're going into I'm going to go into my discussion. So basically, the bacteria was more concentrated at the feeders, and what this can mean is that disease can spread more easily at the feeders, because essentially it's like, if you think about a bunch of people drinking out of the same cup, that's what is happening at hummingbird feeders. And more bacteria spread at feeders, and disease spread more quickly at feeders. And I decided to also study fungi. I was originally only going to study bacteria, but I also studied fungi, and I found that because of this past study done by Jessica G, where she found black fungal growths on the tongues of birds at feeders, but she wasn't able to see if those growths were actually because of fungi. So what I was able to find was that those growths were caused by fungi because there were higher concentrations of fungi at the birds that were at feeders. The reason that this is because fungi is found to grow more in more concentrated sugar, and even with proper maintenance, like changing the water every single day at hummingbird feeders, there's still fungi that grows, which just doesn't happen as often at flowers. But basically, and moving on, the beak and feather conditions were not impacted by foraging habits. I expected there to be more variation in the beak because of that first photo that I showed you guys with the bird that had a very distorted beak, but there were no birds that had heavily damaged beaks that I caught. So in conclusion, flowers are the natural and more healthy option for hummingbirds, and even with proper maintenance with hummingbird feeders, hummingbirds are still infected with fungi. So basically my message to everyone is just to plant more flowers, and because they are lower maintenance than hummingbird feeders, and are also better for hummingbirds in general. So I wouldn't have been able to do my project without these people helping me along the way. So thank you to Fede, thank you to FJ, Naomi, and Frank, and Louisa. So the only times when I went to the hummingbird garden were before it was open in the morning, so there was basically no one else there, because they didn't want to have, they didn't want people to see us listening to the hummingbirds. But I wasn't, yeah.
Do you know what the like negative effects of fungi on their tongues are for
I do not know. I just know what I read in the past study about notes sticking out of other tongues, and a lot of the time the tongues were hanging out of the side of it. But yeah, I don't know the actual. Class 7. Yeah, I can go through the back here.
I was gonna say, you didn't happen to like swab the actual feeder. Did you like maybe as a control?
So, actually, in the last study, they did do that. They actually tested the fungi concentration in the sugar water. But I was just swabbing the
Have you read anything about like the gut microbiome of carnivores?
I don't know that much.
Okay.
I know. Yeah. I mean,
So, everyone loves to go to the hummingbird gallery because there's more and more, like, there's a lot of hummingbirds.
yeah.
You mentioned that they should have better maintenance of the feeders. Would you suggest to remove the feeders, or how would you recommend them to maintain?
It's difficult because hummingbird feeders are really the best way to like. I feel like the fungi grow so quickly that it's so hard to like make sure that there's absolutely none. But I don't know. I think it's not as big of an issue if there's if they maintain it like every day. But it can definitely be a way bigger issue if they're not doing that.
Joe, just to comment briefly, in the direction of Sophia's point, it's interesting to suggest make a swabbing in the feeders and then cultivate that.
Yeah, that would be.
I listened when you mentioned about the tongue of the bee that was not very good. That was in flower context, right? No, wasn't in the feeder.
So the beak that wasn't very good?
No, the beak that was not very good. Which bird was the one?
It was in the feeder.
The one that I showed, the picture?
No. The one that you showed in the picture is in the feeder.
Yes.
But in your result, which is the bird that has lower rank in beak?
Lower rank in beak?
Yeah.
You mean the tongue?
The tongue.
You mean the tongue?
Which bird? Oh yeah, there was a green hermit that didn't have a very. That was one bird that was green hermit that had more fungi and it had.
And that one was not in the feeder.
It was not in the feeders. Yeah.
Okay.
That was like one. I think that was like the one outlier. The rest of them didn't have.
Yeah. Thank you.
But yeah.
Elizabeth, why don't you ask your questions while eating sugar-coated donuts? Thank you.
Are we going to swap it out?
So just some background in case you already don't know about Brachylephrus. The species it affects is Coffea arabica, which is the coffee that's commonly grown and consumed here. And the fungus' scientific name is Familia Vestatrix, but it's commonly known as Brachylephrus for the common host it has. This is actually the beginning of wet season, which is a really important time for coffee producers, as it's the time that all their plants are either flowering or producing fruits. but it's also prime time for the germination of this disease. Oh, sorry, I just realized my notes are on here. It's also prime time for this disease to spread because, as you can see, I listed increased risk for infection, and one of them is increased rainfall, as well as higher temperatures, also depending on how much shade the tree gets, and how much infection is already present in the field. Attached is this diagram of kind of how the life cycle works. So it starts with these spores that attach themselves to the bottom of a coffee leaf, and it will infect the coffee leaf by entering through open stomatas, and it takes about 24 to 48 hours for the germination process to complete. And once it has infected the cells, you'll see these orange lesions appear at the bottom of the coffee leaves that also can have a dusty feel to them, which gives them their common name of coffee leaf rust. So with this, I kind of wanted to look at what conditions were contributing to the severity of this, and I settled on shade and how shade is impacting the severity of coffee leaf rust in my two study sites, Life Monteverde and Finca Florencia. And the purpose of my study was just to add to the research being done on coffee leaf rust, because it's a really important disease that's impacting the coffee production here in Costa Rica, and also just to better our understanding of what conditions are making the severity of this disease worse. So for my methods, I surveyed two farms, Life Monteverde, which is a monoculture where primarily they grow coffee plants, and Finca Florencia, which is a polyculture that has not only coffee plants but also other crops growing around the coffee plants. And these diagrams are actually the two that I surveyed at Life Monteverde. So this was a smaller one, and they used chemical fungicides on this plot, and this was their organic plot, where these were a lot larger trees and these were a lot younger trees. When I entered a plot, I would label the plot with each row with a letter and each corresponding tree with a number. And I would collect things like the size of the tree, how much foliage the trees had, how large was the foliage, how much shade the trees received, how many infected stems I observed on the trees, and how close in proximity were they to other infected trees. I also spoke with farmers to see how they had been mitigating the situation, since it was the beginning of germination and fruiting season for them. And I surveyed about a little over 80 trees. Most of them were from the monoculture, and the ones from the monoculture tended to be, well, at least in the organic plot, they tended to be a lot larger, and the ones in the chemical plot were a lot younger, and then the ones from Finca Florencia, the polyculture, kind of varied from medium to larger trees. And I created this graph to show how many trees there were per infected stems, and they're color coordinated to represent the shade ranges they were found in. So anything in full shade ranges were those that were 75% shaded or more throughout the day. Partially shaded areas had about 50% shade range throughout the day, and those labeled none had 25% or less shade throughout the day. And I tracked the sun by southeast, or rises in the, no, that was wrong. Rises in the east, sets in the west. Question mark? Okay. And so yeah, I created this graph, and I found that from my results that there was the least amount of infected stems were mostly in the sun, and the most infected stems were in partially shaded areas, while the table represents the average of infected stems by the shade range, and again, partially shaded areas had the most with an average of 6 infected stems, while areas with not that much shade had an average of 3 infected stems, and fully shaded areas had an average of 2 infected stems, as well as the fully shaded areas varied in how many infected stems they had, so it could be from 7 to 0 infected stems. And from my results, I concluded that maybe the reason that partially shaded areas were the most infected is because Majority of them came from the monoculture, and in the monoculture, the trees tended to be a lot closer in proximity to other infected species compared to the polyculture, and also because they had a lot larger leaf foliage, and the disease doesn't tend to infect new leaves and new trees. So yeah, in conclusion, shade is not influencing the severity, but the spread of the disease, kind of how like it spreads with wind and rain splash. And in the future, I would want to do more research on how the foliage size, the spacing between the trees, and the proximity to the infection is what's increasing the severity of the disease in the species. Thank you to both farms, like Monteverde and Finca Florencia, for allowing me to go do research. Thank you to my advisors for helping me with my research. Thank you to UCEAP, Estación Biológica, Monteverde Institute for giving me the resources for being able to do my research, and my homestay family for giving me really good food when I was counting trees all day at the farm. Without all you guys, I would not be able to complete my work.
We have time for a few questions.
I would want to look more at mitigation efforts because I'm really interested in like organic methods of mitigating the disease. But also now that I've done this research, I would want to look at how spacing and size of the leaf foliage is going to influence the spread of the disease.
Do you know if there's any like preventative measures that farms can take to prevent the disease?
Currently, like chemical or organic?
Just either way.
Chemical, there's like a lot of fungicides that they use, but then that prevents the farm from being able to call itself organic. So like at Finca Florencia, when I spoke to Mauricio, he was saying that he used a combination of like ash and a sulfur, and he removes his fungus that way, but there's also just premium. But my recommendation would be to grow the trees a lot further apart, because even just looking at the monoculture versus the polyculture trees were a lot further apart, and so the disease wasn't like spreading over the whole plot, whereas in the monoculture, it seemed to spread a lot more just by how close in proximity the trees were. Yeah.
Sorry if you mentioned this and I didn't hear it. How does it spread? Is it by wind or?
It spreads by wind and rain splash. That's like the main way it like spreads, and then it's able to germinate. And things like increased rainfall, higher temperatures, the extent of the shade, or the proximity to other infections is what causes the severity to be a lot worse. Yeah.
What exactly is the severity of the fungi to the amount of fruits produced? Like, does it completely, how exactly does it do that?
Yeah, so what the disease does is once it infects a leaf, the leaves are going to fall prematurely. So if it infects enough leaves, all the leaves are going to fall off the tree, and the tree is going to try to keep growing leaves, so it's not going to put all its energy into the fruits it's supposed to be producing, so then it produces like bad fruit or bad coffee.
Okay, thank you very much. So we have some serious other things to do, including taking a picture and then eating donuts and other things, and then Naomi's going to arrange for transportation.